Welcome again to our day 2 of our webinar on enhancing vaccine efficacy, unlocking the power of adjuvants in global vaccine development. We had a very nice session yesterday with Dr. Derek, Julie, and Mark. So today we continue with more presentations. Hannah, can we move to the next slide? Again, for those who didn't join us yesterday, again, briefly, we just wanted to mention what the scope of our webinar was. It's intended for LMIC manufacturers so that this can provide an overview of vaccine adjuvants, different vaccine types, the immunology, and the mechanism of action. So this may help to develop new vaccines that need adjuvants and how one could modulate that in CMC for manufacturers. A little about PATH, who have not been here yesterday. We are a global nonprofit organization in the health space. We are located about 70 countries all around the world. And our goal is to make health easier and accessible to low resource settings by providing affordable health products, including vaccines. We are a part of the PATH's Sustainable Vaccine Manufacturing Program, which is sponsored by the Gates Foundation. And our goal here at the what we call SVM is to provide technical assistance to LMIC manufacturers to enhance their abilities and to develop and sustainably produce safe, effective vaccines so that they can be of international quality and meet all the public needs. So on the next slide. The logistics again, I repeat that the participants all remain muted with your webcam off. Note that the webinar is being recorded. Please do not record. We will provide you the recording in June. If there are questions, please put them in the chat, and we will only use the chat feature for the questions. And that makes it easier for monitoring all the questions and responding to them at the end of the talks. Your questions are being monitored and we will collect them. And if we are not able to get to the questions after the speaker's talk, we'll definitely try to field them after the webinar today. And also, if we still don't get to it, you're free to email us and we'll make sure that you get your answers. After the webinar, we will also send a short survey to get your feedback, because this feedback will be very helpful for us to be able to understand how it has helped or made an impact on the LMIC community. And this will help us also to bring more of such webinars in the future. The presentations again are going to be recorded and they will be publicly available on the PATH website. The next slide, today's agenda. We have 4 speakers today, and we'll start with Dr. Daniel Fullen from CEPI. Then we go to Dr. Dennis Christensen from CRODA. Then Dr. Wolfgang Littner from NIH, and then last but not the least is Dr. Jay Evans from Inimmune. So next slide. Before we start, a little brief about Dr. Daniel Fullen. He is the lead for R&D innovations at CEPI, where he leads the CEPI strategy and portfolio for vaccine adjuvant innovation. His work focuses on expanding global access to adjuvants by addressing technical intellectual property and manufacturing barriers and enabling equitable use in middle-income countries. At CEPI, he oversees the adjuvant library, designs and manages calls for proposals for novel and existing adjuvants, and works with academic, industrial, and public sector partners to support evaluation. So with that introduction, we will request Daniel to take over.
Thank you. Yes, so thank you for the introduction. So as mentioned, my name's Dan Fullen. I've been at CEPI now for 4 years, I believe. I'm a virologist by training. Yes, and I lead the adjuvant portfolio at CEPI. So today's presentation, I'll give a brief introduction of background to CEPI for those that are not aware of CEPI, and then I'll lead on to some of the work we're doing on the adjuvant library and where we're intending to go with that into the future. So next slide, please. So yes, so CEPI was founded in 2017 off the back of the Ebola outbreaks that happened in 2016. And 10 years later, obviously, we have another major Ebola outbreak going on. In that time, there's now an Ebola Zaire vaccine available, although there isn't one for the new emerging strain, and a lot of work is happening at pace to do that. The remit of CEPI was really to make sure that these things couldn't happen again, or to mitigate as far as possible should they happen again. In the Ebola space, I'm not an expert in this field, but a lot of CEPI's work over the last 10 years has been clinical trial support, in running and supporting clinical trials in the events of Ebola outbreaks, for example, supporting early vaccine development as well. And there's a lot of work going on, obviously, now for the new outbreak, and also to support the advancement of early stage candidates and potentially a filovirus vaccine in the future as well. Next slide, please. So CEPI is a global partnership of countries from around the globe. The purpose of CEPI is to try and support or help create a world where epidemics and pandemics are no longer a significant threat to humanity. And part of this role is to help accelerate that development of vaccines and biologics as well, and to ensure that they're accessible to people everywhere where they're at need. There is the mantra, no one is safe until everybody is safe. Next slide, please. So yes, as I mentioned, how do we make everyone safe? These are through improving equitable access conditions, manufacturing supply, also measures such as thermostability as well, and improving access terms to technologies that are already available. So this diagram here just shows where CEPI fits into the current global landscape. We're very much focused on R&D, so we're trying to push a lot of R&D, new innovations through vaccine innovations and technologies. We're looking at mechanisms where we can try and streamline regulatory policy or shape that in peacetime, so that when there is a pandemic or an outbreak, some of those barriers have already been broken down and we can implement these new vaccine technologies in a quicker manner. CEPI is also looking a lot at manufacturing capacity as well, either improving manufacturing at scale, but also enabling more regions as well to have that manufacturing capabilities and capacities. So there isn't the dependence on just one or two suppliers for the whole world, but also bringing down cost of goods and things like that as well. Next slide, please. So CEPI instigated the 100-day mission. The purpose of the 100-day mission, I think there's one more click there, please. Yep, that's the one. So CEPI instigated and founded the 100-day mission. And this is the idea and the aim that we could potentially be developing vaccines from a new outbreak to 100 days and putting all the measures in place that are required to enable us to manufacture a vaccine at scale. And these are the measures from innovations, regulatory innovations as well, and that manufacturing at speed and at scale, but also by having these new vaccines developed within peacetime so they are available when these new pandemics and outbreaks come along. Next slide, please. As everyone's very well aware, during the COVID pandemic of 2020 to 2022, the COVID vaccines were made at record time, a year, which had not been seen previously to bring a new vaccine online. What has been projected, however, if we are in the future able to develop a vaccine within 100 days, taking COVID, SARS-CoV-2 as an example, it had been projected that we could have saved potentially up to 8.3 million lives by being able to make a vaccine in 3 months rather than a year. Although what was done before was a huge leap forward, we feel we can go further in the future. And CEPI's mission is to try and get all these measures in place to make this more of a reality going forward. Next slide, please. So this year, we're approaching the end of CEPI 2.0, and we're now making the ask for CEPI 3 or CEPI 3.0. This is a 2.5, the idea is a $2.5 billion strategy for the next 5 years of CEPI. A lot of the focus of CEPI 3.0 is going to be on the viral families. So these listed here are the viral families that are anticipated to be the cause of a pandemic or major outbreaks in the future. We'll be taking, representatives of these families to try and develop vaccine candidates and get technologies in place around these so that we have a standing start for these outbreaks. So as you can see, just the main families here and some of the disease indications on that. Next slide, please. So changing tack, more onto the topic of adjuvants, the scope of these webinars. So I just thought, first of all, I'd do a recap of some of the discussions yesterday and those that hadn't attended as well. So next slide, please. So as I'm sure you're all aware by now, an adjuvant is a material or a substance that really helps boost the effectiveness of a vaccine. This can be by tailoring the immune response, either strengthening an immune response or tailoring it to more of a T cell, a B cell response, or a balance of the two as well. They can also help by improving the durability of a response as well, potentially reducing the need for booster doses as well. They can also have the benefit of dose sparing, so reducing the amount of antigen that may be needed in a vaccine as well. Next slide, please. So what makes an optimal adjuvant? So the next slide, please. The true answer to this is it really does depend. Adjuvants are really context specific. So depending on who you want to vaccinate, that's your target population, a different adjuvant may be required. This could be for different adjuvants may be required for a pediatric population versus an elderly population, obviously naive versus pre-primed as well. And then other considerations are needed again for people that may be immunocompromised. The type of adjuvant also key, and most importantly, depends on that type of immune response you're looking for, which is also related to what immune response do you need to target a particular pathogen. So that could be the efficacy and strength of that response. Is it an acute immune response you're looking for? Or is it a breadth, to cover many different strains like you may see with flu, for example? And then duration of response as well. Is a year enough? Do you want protection for longer than a year? And then other considerations, sometimes, but not always, linked to the immune response is the reactogenicity as well. The route of administration is also affected. What adjuvant you use, you may use one adjuvant for an intramuscular vaccine, but if you want intradermal or intranasal, it would be a different one. And that also will affect the immune response and the outputs there. And again, the platform as well. RNA adjuvants are not really to the fore yet, but may benefit from those in the future. Depending on the platform and the delivery, coupled with the route of administration, will also affect the adjuvants you select. And as I mentioned, depending on the pathogen, the type of immune response will be different again. So if it was an acute infection, you probably want an antibody-driven response. But if it was something more chronic, like malaria or TB, a T-cell response may be more of a priority. It's also important to consider as well the CMC aspects of that. So the cost of goods are really important, and dose sparing potential also would have an impact on the overall cost of a vaccine. That has to be balanced on obviously the immune response. Something like a component such as QS21 is quite high in cost, which will increase the cost of a vaccine, however, will also enhance the T-cell response. So those kind of balances have to be taken into consideration. And then there are also the more political aspects as well, and less scientific considerations when selecting a vaccine. This could be IP. Are there restrictions on use for certain adjuvants for certain disease indications, or in certain territories? Can you get access to that adjuvant full stop? What do the licensing terms look like? Is it an open access adjuvant? Again, restricted IP terms. And the cost of getting a license to that adjuvant would be a consideration. There are also wider geopolitical contexts as well. You know, is an adjuvant produced in China or the US? That sometimes factors in, although that shouldn't really matter. But those, we can't be blind to global politics as well as to where these technologies are available and come from. Next slide, please. And just to reiterate, as mentioned yesterday as well, and I think is also a very important consideration, adjuvants do operate in what I would say is a regulatory grey zone as well. They are classified as excipients, but they do have a biological effect. So they're not an excipient in the truest sense. They're not inert, but it is really dependent on the adjuvant and how that immune response is directed on the antigen. Apologies. Very loosely, the EMA would classify an adjuvant as an excipient with active-like effect, and the FDA have essentially called it a component of a licensed biologic. And on that, it's really important to also consider that adjuvants can't be licensed in isolation. Their effect and output are dependent on the combination of the antigen and the route by which it's administered. So that does make it more difficult to bring them forward into a wider acceptance. As a part of that, that does complicate things in terms of that access as well. So that leads us on to the next slide, really. And I would say, so next slide, please, the wider adjuvant problem. So the next slide will just highlight some of those key issues, really. So adjuvants that have been used in licensed vaccines are in high demand because they've been seen to be de-risked. They've gone through that regulatory scrutiny and have been used in a licensed product. However, that is specific to that particular disease indication or that antigen. If an adjuvant has been licensed in a influenza vaccine, its behavior with an Ebola vaccine, for example, may be completely different, and it should really be seen as a new product. However, that is not the general perception. Because adjuvants used in a licensed vaccine have gone through that regulatory scrutiny, they have been de-risked, that does add a lot of value to them. As a consequence of that, there have been a lot of mimics being developed that have a similar composition to those used in licensed products, again, have been seen to be de-risked. As a consequence of that, there's a lot of value added with these licensed vaccines, which makes them less accessible because either big companies may have them use them only for their own products and don't want to risk them being used outside of their control, but also that value added makes them very expensive to access as well. Because they're context specific, it's not always clear what is the right adjuvant to choose. And because of those restrictions on IP and access terms and price, many vaccine developers will pick an adjuvant that they can get their hands on rather than any rational selection. It might not be the best adjuvant for their vaccine, but it's what they can get access to. As we saw during the pandemic as well, the SARS-CoV-2 pandemic, lots of countries closed their borders. There was the US Defence Production Act, and the EU as well looked after itself. So as a result of this, those materials were not getting to where they needed to be. And that leads to a bit of a gold rush for either raw materials and these adjuvants as well, particularly the global south, so many times seen back of the queue. Another really important consideration as well with adjuvants is when a vaccine developer picks an adjuvant, they may only get a license to use it for preclinical or phase 1 or phase 2 development. And if the adjuvant developer, for any reason, withdraws access to that adjuvant, you're back at square one. you really have to start again. You have a new vaccine. So that work you've done in phase 1, phase 2 will not apply if you have to change your adjuvant. You're back at the beginning. So it's really important, if you select an adjuvant, as far as possible, to negotiate access terms up to, or conditions up to, a licensed product. The other condition is if the adjuvant is working well in phase 1, phase 2, and you then want to negotiate access to that adjuvant for phase 3 or beyond, you're very dependent on that, and you may get less favorable terms financially to take your vaccine through. You're very dependent on that. So next slide, please. And these circumstances are not getting better. Because of the success of particular adjuvants, they're getting more expensive. Novavax with their Matrix M adjuvant is in a lot of demand, as you can see here. Sanofi did a 1.2 billion deal for access to Matrix M, and also Pfizer as well has taken on a big deal as well to cover their programs. Dynavax was also recently acquired by Sanofi. Dynavax, the producer of CpG 1018. It's not clear how accessible that will be for the future for vaccine developers. So again, as the market gets more constrained and some of these technologies are acquired or tied up in big money deals, they become less available potentially for other vaccine developers. Next slide, please. So at CEPI, there are many different angles. We're trying to address some of these issues and try and improve accessibility of adjuvants, but also to move them forward and support vaccine developers. So what we've done over the last couple of years, we've developed an adjuvant library. And this is a physical adjuvant library of 25 adjuvants from 14 vaccine developers. We originally put out a call. We had over 70 applications to the library, and we down selected those to these group of adjuvants that were best suited, we thought, for public health needs. These adjuvants were selected on a number of key criteria, as in they are all suitable for bedside mixing. That was a practical consideration. All the developers were willing to partner, in principle, with vaccine developers, should they be selected and partnered up as a result of the adjuvant library. They can produce at scale, their adjuvants at scale, or have a partner that can potentially produce, they can either manufacture their adjuvants at scale or have a partner that could do that on their behalf. And also all these adjuvants have been tested preclinically as well, as a minimum. Next slide, please. So I'll come on to in a moment how the adjuvant library runs and how people can access that. But in principle, the benefits of the adjuvant library benefit vaccine developers and adjuvant developers as well. So for vaccine developers, it enables broader access to adjuvants that they may not have either been aware of or been able to access. So we have a diverse range of adjuvants across the major classes, and vaccine developers can come to us to request access to this. The adjuvant library process is to enable a more rational adjuvant selection based on your disease target, on your immune response needed, rather than just what you can get your hands on. The vaccine developers will also get preclinical data on how their vaccine performs with a range of adjuvants, at an immunological level, but also in a challenge study as well. We also hope, by improving access to these adjuvants and making more available, it will improve market competition in this field and maybe have a downward pressure on overall cost of goods for these key materials as well. Next slide, please. Also, obviously, we're very grateful for all the adjuvant developers that have contributed to the adjuvant library to date. And we also hope going forward, as it becomes more established and we ramp up the turnover of studies, that we have a lot of benefits to adjuvant developers as well. First and foremost is by introducing vaccine developers to the adjuvant library, it gives their adjuvants greater visibility that they may not have had beyond the bigger brands that are out on the market and very expensive. So by connecting them, we hope also it improves their exposure. It also helps the adjuvant developers. As I mentioned, they're not licensed in isolation. So if the adjuvant developers are partnered with more vaccine developers, they have more shots on goal. So it increases the likelihood of their adjuvants moving down that developmental pathway, phase 1, phase 2, phase 3, and potentially into licensed products, which adds value to their technology. By participating in the adjuvant library as well, the adjuvant developers will get the preclinical data on how their adjuvant performed against a range of pathogens as well, which they can use to build trust in their adjuvant and also use for regulatory submissions in the future too. Next slide, please. So this slide here just shows an overview of what we currently have in the adjuvant library. We've grouped them very loosely into the major classes. So at the top here, you can see we have adjuvants from MaxVax, and Mark spoke about this yesterday, some of their portfolio. The liposome-containing adjuvants with the QS21, generally very good for a T-cell response. And they're ranging from adjuvants that have been produced as GMP materials, but may not yet have been used in a clinical trial. My apologies if these aren't the most up-to-date stage of development, but this is the data I currently have. So we have MaxVax with a GMP material, but also their MA105 they mentioned in phase 3. ARHI with a TLR4 agonist liposome, and RecBio's BFA01. Then we have the squalene emulsions containing vitamin E or alpha tocopherol, very similar to GSK's ASO3 or mimics of. Again, we have MaxVax there in a GMP grade, right through to those that have been used in licensed products, such as Shionogi's adjuvant there and RecBio's BFA03 as well, also been used in licensed vaccines. We have one TLR4 agonist bound to an alum, which is the GLA alum from ARHI, and the other alums, obviously, can't forget the alums from Croda as well, which have widely been used in licensed vaccines as well. We have a couple of CPGs from Par Bio in China, and the emulsipan from Panacea, which is a similar composition, I suppose, to the MF59 adjuvant, and that's been licensed in their Pandiflu vaccine, I believe. And then lastly, we have our other group, which quite often is the most interest to academics because there are lots of more novel compositions here, as you can see. And the one used in the licensed vaccine is the MCT from Allergy Therapeutics, which was licensed in their pollen vaccine, I believe. And as participating in the library, we hope to move more of these adjuvants from the left more to the right-hand side, or at least to be able to help that process as well. Next slide, please. So how does the adjuvant library work? So as a vaccine developer, you can come to CEPI and have a discussion with us about using the adjuvant library. It's, as I mentioned, it's a physical library, and that's stored at the MHRA labs in the UK, NIBSC. A vaccine developer can come to us. You can pick up to 5 adjuvants from the library, or 6 if you don't have an existing formulation. What we would do is, on request and discussion of the composition of the adjuvants you'd like to test, we would then ship 5 adjuvants to the vaccine developer. They would then conduct their own in-house compatibility testing. We let them do that because the vaccine developer is the expert with their own antigen. This is very high-level compatibility testing, Western blots, ELISAs, things like that. Some short-term stability studies up to about 8 hours. And that's just to confirm that the buffers or the adjuvant wouldn't denature the antigen and that it won't lose its efficacy before committing to any in vivo studies. When the vaccine developer is able to confirm compatibility of the adjuvants with their antigen, we would then ask the vaccine developer to ship their antigen and also existing adjuvant formulation they may have to one of CEPI's animal model network laboratories. Now, these are a series of labs around the world that have been pre-authorized and assessed by CEPI that can conduct studies on our behalf. So the vaccine developer would ship their antigen to one of these laboratories. We would then ship the 5 adjuvants from the adjuvant library to that testing site as well. We would then conduct bedside mixing with those adjuvants and then run a head-to-head comparison study, initially in a mouse model in black 6 mice. Then, as a result of that, the typical readouts of that study would be neutralizing antibody titer, T cell responses as well. And we're also looking to build other assays in there as well, maybe looking at CD8, IgA in the future as well going forward. On the basis of that study, the vaccine developer gets all the data on that study. The adjuvant developer will also get all the data relating to their own adjuvant. The adjuvant developer would also get semi-blinded data as well. So they would see how they compared in the head-to-head comparison study of those 5 adjuvants, but they won't know who they've been compared against, because obviously adjuvants are context specific. In one study, there's going to be an adjuvant that outperforms the others, and there may be one that comes last in that particular comparison study. And as you can imagine, if you became top of the ranking, you probably would like to tell everyone, but if you don't in that particular study, you might not want to. So that's why we have to very carefully handle that comparative data. On the end of that study, the vaccine developer would then pick the lead 2 adjuvants they would like to take forward into a challenge study. So this would be a disease-specific challenge study. Again, be run to a similar format. Based on that study, we would then hope the vaccine developer has a lead adjuvant candidate they would like to take forward. CEPI would then step back and let the vaccine and adjuvant developer negotiate terms. And the only condition on our behalf is that any deal that is done is on a non-exclusive basis. So if it's a great adjuvant, we still want that to be available to other vaccine developers as well and not taken out of the market, because that's one of the key problems where we're trying to solve. Also within CEPI, we're looking to build an adjuvant database as well. So these studies are run to a standardized model. So the idea is that over time, we can cross-compare studies, and we may be able to shortlist in the future which group of adjuvants would be the best ones to look at based on your target antigen, route of administration, or the vaccine platform that's being used as well. And over time, as that builds, hopefully that will become more valuable and useful. Next slide, please. So this is just an overview of how one of the mouse immunogenicity studies would be conducted. It's a typical prime boost study. And as you can see, we have the 5 adjuvant groups there, conducted in black 6 mice, compared against the current adjuvant control, which will be the vaccine developer's existing formulation. We're also using ADA-SO3, which is similar or mimic of ASO3 as a comparator as well. And we use that in every study to help us normalize the data and also cross-compare across different platforms as well, and an antigen-only control. Next slide, please. And this slide here is just an example of one of the challenge studies. This is just taking SARS-CoV-2 as an example. It may be a different animal model, very likely, depending on the disease indication to go forward with as well. Next slide, please. So as I mentioned, within CEPI, we'll be looking to also build an internal database of this. So this will, as I mentioned, help us to analyze the data from individual studies, hopefully look at how different adjuvants will perform across different disease targets or vaccine platforms as well. Within CEPI as well, and this is a wider piece, even beyond the adjuvant library, within CEPI, we're looking to build a pandemic preparedness platform. And this will be everything from antigen design, clinical trial data, adjuvants as well. So we hope to feed this adjuvant data into this AI system, where in the future we can fast track pulling many different parameters together to help identify or predict what may be the best vaccine formulation for any disease X platform coming down the line as well. So this data will all form part of that. Next slide, please. So yeah, just to summarize, so over in the horizon, it's still relatively early days with this vaccine library, with the adjuvant library, and we very welcome people coming and approaching us to use it. Obviously, CEPI's focus is viral pandemic pathogens, but the adjuvant library is also available to people developing vaccines in other areas as well, malaria, TB. We probably couldn't cover the cost of a non-viral vaccine, but we can certainly give access to the library. So yeah, if you have something else that's outside of viral pathogens, please do come in and talk to us about it. So in the not too distant future, we've just finished our first SARS-CoV-2 studies. We're now looking to move in doing Nipah, SARS-1. We have some more SARS-2 vaccine candidates, some H5N1 and pandemic flu vaccine candidates we're going to be testing, and some pan flu vaccines as well, MERS, and also Mpox as well in the pipeline. So any adjuvant developers on the call that are patiently waiting for that data, these are coming, and hopefully we'll be able to share some of that information with you in the not too distant future. Next slide, please. So where next? So it took approximately 2 years to get the library up and running. One of the big unique things was that we've been able to get to an agreement with many adjuvant developers that all signed the same contract to enable us to cross-compare. Now we're starting to get that data. Hopefully that will snowball throughout this year. We're also looking at where we could go with the adjuvant library in the future as well. We're starting to lay the groundwork for mucosal adjuvants. We're going to be incorporating IgA assays into the adjuvant library. And hopefully over the next year or two, we will be asking for specific mucosal adjuvants to be submitted to the library as well. These will be adjuvants that could be IM and elicit a mucosal response, but could also be adjuvants that are administered mucosally as well. So how that looks in the future will be really good using this framework. As I mentioned as well, we have this pandemic preparedness engine that has been developed at CEPI. We'd like to use AI support as well to help with that adjuvant selection and accelerate those processes in the future as well. A bit further out, but what could be really interesting, using the adjuvant library framework to maybe build an adjuvant library specifically for RNA adjuvants. These could be head-to-head comparisons of LNPs potentially, or it may be an additional adjuvant added to an existing formulation, maybe delayed release as to not shut down translation, but also maybe, enhance and prolong the immune response of RNA vaccines. RNA vaccines, as you know, are very good for rapid response, but typically have a relatively short duration. So if adjuvants can help in that space, that would be transformative. Also, finally, as we have a standardized protocol and a standardized system for assessing these adjuvants and vaccine candidates, we'd like to also potentially use this as a tool to assess emerging technologies. So this could be organ on a chip or lymph node on a chip, could be compared to the existing in vivo models. And also we may be open to groups with in silico analysis to use this in parallel as well. So next slide, please. I think that was the last one. I'm happy to take any questions.
Thank you, Dan. That was really very helpful to see all the CEPI library and what one can actually do. I think questions, the first questions, what are the adjuvants that you would recommend for mRNA vaccines, especially like malaria or TB?
So to date, there aren't that many that are coming to the fore. I think there are a few groups in Korea that have been developing some. I can't remember top of my head, but I think one of the key things with RNA vaccines is because you have that translation, what has been seen in the past is that early innate immune response can shut down translation. So we're the really interesting field, I think, beyond LNPs, which could be classified as an adjuvant, where the really interesting innovations are delayed action adjuvants that are co-administered with the vaccine, but they don't activate until a few hours later. So you've had time for the RNA translation to start, and then a delayed innate immune response. So that's, I think, where the innovations are going to be. It could be really interesting.
Yeah, thank you. We have more questions.
Mhm.
Robert, do you want to field them?
Sure. Yeah. Thanks, Lakshmi. Okay.
Mhm.
So yeah, Dan, this is great. Thank you so much for this. I think there's a few questions I'm seeing related to the AI software that you're using, and kind of more generally about what you see as the, if you could elaborate more on the role of AI in, like you mentioned, adjuvant selection and so forth. So can you give us any more details on that?
Yeah, so that's my colleague Newton Wahome leads the AI platform at CEPI, and the grand ambition is that this is an all-encompassing system. I think they're working with Argonne Labs, and I've been talking to some of the supercomputer sites in Singapore and the US in particular. So this will be everything from antigen design to pandemic mapping. We may be looking at in the future doing some more. At the moment, it's all bedside mixing work, but we may bring on a facility that can do more in-depth formulation, looking at biophysics interactions between specific antigens with particular adjuvants as well. That's something potentially for CEPI3. The data we get from the adjuvant library, we hope to feed in there, as I mentioned, so we can accelerate those predictions of what would be the best immune response for an antigen or a certain disease indication as well. So it's still early days, but yeah, we hope just to fast track everything with that potential approach, really.
Great, thank you. And then the next question is if there's plan to add new adjuvants to the CEPI library. So can you maybe talk a little bit more about how, what you're thinking about that process for that?
Yeah, certainly. So we are really keen to expand the adjuvant library. It will go through a natural curation of how things are being used and bring new things in where we identify gaps as well. At the moment, where we are, we're not bringing in new adjuvants into the library at the moment because the first members of the library, the vaccine developers, have been very patient with us as we got everything up and running and get all the contracts in place. So what we want to do initially is the adjuvant developers that are already in the library that went through the first selection process, we want to give them the first exposure to the first round of vaccine developers that come in. So when they've all had a first decent run out and the first adjuvant developers to the library have had the first exposure, we may launch a new CFP, realistically sometime later next year, to bring in a new tranche of adjuvants into the library. One thing we are currently doing, because the existing developers have already signed the contract to participate in the library, if they have new adjuvants they'd like to add, they can do that. That's under the existing agreements as well. But as things evolve and we identify those gaps, we will look to bring more in as well.
Great,
So hopefully next year we'll have that, particularly in the mucosal space, I think.
Great, excellent, thank you. I think we just have time for maybe one more question before we move to the next speaker. There's a question about the animal study. So I know you mentioned briefly mentioned the lab network, CEPI lab network, but if you could say a little bit more about that, thank you.
Yes, certainly. And I was going to incorporate some slides into that, but I wasn't sure if I'd have time. So the animal model network that we have is approximately 20 laboratories from around the world. That's the US, Europe, South America. We're bringing some on in Africa now as well, and Southeast Asia. These labs have all been vetted for their capabilities, and they have labs from BSL-2 up to BSL-4 as well, and a range of capabilities. So we've been supporting some of those labs to help upgrade their standards. So the 3 Rs of best practice on animal husbandry and care as well, and high containment as well. And what we can do is we can introduce vaccine developers to those labs. If they're looking for certain capabilities and assays, we can support with that. But we can also influence the direction of travel with that as well. So in some of our challenge studies, as you probably saw there, we're going to be having a NEPA challenge. That will be BSL-4. So that's one example of one of the studies there in the high containment labs that should be due to start within the next couple of months, those studies.
Great, excellent, thank you. I think we need to move on to the next speaker.
Yes.
So thank you,
Okay.
Dan, very much. And if there's other questions for Dan, please put them in the chat and we'll try to get to them at the end after all the presenters, if we have some time at the end of the day today. So Lakshmi, if you could go ahead
Yes,
and move
yes.
or
Thank
introduce
you.
the next speaker,
Yes.
please. Thanks.
Thank you. Thank you, Robert. Our next speaker is Dr. Dennis Christensen from CRODA. And a little background about him. He's the head of global research and development at the adjuvant systems for CRODA. And with he's been leading the effort to expand CRODA's offering of new and innovative vaccine delivery systems, immunostimulators, and combined adjuvant systems. He has more than 20 years of working in the pharmaceutical and immunological aspects of vaccine adjuvants and delivery systems, including targeted delivery vaccines. In this period, he has played a key role in the development of the CAF series of adjuvants from Staten Serum Institute, of which the CAF01 and CAF09 are now being developed as commercial products by CRODA. He's a key opinion leader in the vaccine adjuvant space and has an extensive experience with international vaccine research and development programs. With that, Dennis, please, we welcome you to present.
Thank you very much, Lakshmi, and thanks for giving me the opportunity to present at this very important webinar. Yeah, just briefly, I'm representing here as head of global R&D for vaccine adjuvant systems, but I am also holding a research professorship at Staten Serum Institute. So having a foot in both camps. So if you can move to the next slide. So during this webinar series, you've heard many different versions of what vaccination is and what role adjuvants play, and there will be a lot of repetition. But I also heard that people need to hear things 60 times before they understand it. So you might have a chance after this webinar to have a better insight into vaccine adjuvants. I usually say that vaccination is all about presenting sufficient amount of the right antigen in the right conformation to the right cavity in the right cell population while supporting with the right co-stimuli for a sufficient amount of time. So if you fulfill these 5 points, you actually have a pretty good vaccine. Of course, there's a lot of question marks associated to these, but if you just press to the next. But actually, if you group these 5 points, you could say that the antigen really plays a role for the first 2 points with the right antigen in the right conformation, while the adjuvant actually plays the major role in the delivery and the co-stimuli aspects of it. And therefore the adjuvant plays an enormous role. And that's also the reason why it's really important to bring in the adjuvant very, very early in your antigen discovery programs, because these two things need to grow together into your final drug product. If you go to the next slide. So I think you have seen various versions of this before, that we had very early on in the 1930s. We had the first introduction of the aluminum adjuvants. They were developed by Gaston Raymond, who had the task of improving the anti-diphtheria antitoxin antibodies production in horses, and basically mixed whatever he had on the shelf with his diphtheria toxins and injected them into horses, and actually found that alum was a really, really good adjuvant. And only very shortly after, the first vaccines were introduced into humans, including aluminum adjuvants. I think the first one was in 1936. Then nothing happened for a very, very long time until in the mid-90s, where especially I would highlight the MF59 was introduced. Derek talked a lot about that yesterday, or that family of adjuvants yesterday. So I don't want to go much deeper into it. Just saying that after that period, we have had a number of important introductions of novel adjuvant systems, not least here in the last few years, especially due to the COVID pandemic. But also really with having big farm companies like Novartis and GSK, Dynavax, Novavax, being maybe a little smaller, but introducing new adjuvants into the field. And I think that's really important to have in mind, because if you go to the next slide. That also means that and that was also what Dan said, that these adjuvants are really not available generally for other vaccine producers. It's not the task for GSK to supply adjuvants to the rest of the vaccine producers in the world. But the problem is, of course, for others is that how can they then get access to vaccine adjuvants? If you go to the next slide. And that's where we in Croda are aiming at filling that goal. Our purpose within vaccine adjuvant systems is really to design and develop vaccine adjuvants for any purpose needed. That's a quite ambitious goal. I'll come back to how we do that later. We want to mature these vaccine adjuvant technologies by developing manufacturability and scalability. So we are really focused on very early on in our inclusion of novel adjuvants to also focus on process development, CMC, scalability issues. We want to make novel vaccine adjuvants reliably available to vaccine innovators, meaning that if you engage with us, and you can also be sure that they will also be available in commercial scale in the future, even if it is novel adjuvant systems. We want to supply both developmental and commercial volumes in line with our customer readiness, meaning that well, of course, we are full aware that in the development process, you have various different needs in volumes and scales, and also technological services, technical services from our side, and we want to grow together with you in that collaboration. And then we want to make them available to the world, not only to certain selected partners, but actually making them broadly available, completely in line with what also CEPI is focusing on. Next slide. So our portfolio is quite broad, and when we say vaccine adjuvants, we think a little more broad, because we both focus on establishing the infrastructure and supply chain for the building blocks, for the immune stimulators, and then the final systems. And I'll in this presentation focus a little more on how we do that. If we go to the next slide. So for each of these categories, our strategy is really to ensure a bench-to-bed infrastructure, so that we can take new products from early research all the way through into commercial production, ensuring the supply chain as needed on that path, and also ensuring the quality specifications needed, going from early research where maybe the COA is not so detailed, all the way into a final product where there is a very solid documented data package associated to it. And we grow that in the development of our novel adjuvants and excipients. Let's move to the next slide. Yeah, next slide. I wasn't aware that I didn't control this myself, so there's a lot of intermediate slides here. Focusing first on the building blocks. So in all modern adjuvant systems, that is always associated a number of building blocks. In many of the novel ones, it is based on lipids, on oils, surfactants, and so. If you go to the next slide. So part of our portfolio is that we a few years ago acquired a company called Polar Lipids, who has where we have access to more than 2,000 unique research catalog items in the lipid family of molecules. With that, we also have an infrastructure that enables us to take these research items into GMP in small scale, do the CMC, and then grow it into our wider Croda production facilities all over the world to produce these compounds in large scale. Go to the next slide. Quite slow. So within that portfolio, and also expanded a little afterwards, we also have a number of different immune stimulators. In this presentation, I will only focus on a few of them. So if you go to the next slide. So one of them is our synthetic MPLA analog and analogs, and these analogs are really as close as makes rationally sense analogs to MPLA. MPLA is not a single component; it is a natural composition of a number of different molecules with our series of different analogs. We make them synthetically, and therefore they are more pure products. And we have 3 different versions in our catalog, all in GMP. We have the FAT, which is our first generation. It's also referred to as GLA from AHI, formerly known as IDRI, called it GLA, but it is the FAT molecule from in our nomenclature. We have 3D FAT and 3D 6-acyl FAT, which have various degrees of freedom to operate, but we can for sure find one that has the where there is accessibility. They are all potent TLR4 agonists, and therefore inducing a strong Th1 response. And they have all been tested in multiple clinical trials. If you move to the next slide. Another example of how we operate is also that we look into the general world supply chain and say, where is there a need, and where can we, with logical, rational, with our expertise, take in products and make them available in a better version. One example is the squalene, which is effectively used in many different adjuvants. Also, most well known, of course, the MF59 and the ASO3 adjuvants. These adjuvants contain the squalene as the critical immune stimulator. It's a damp signaling molecule. You heard about it yesterday with Derek's presentation. The issue so far has been that the supply comes is extracted from liver oil from deep sea sharks, and of course that creates a problem both environmentally. Of course, the sharks do not like it, but also from an environmental and sustainability point of view, it's not a good supply chain to have. If you move to the next slide. Therefore, we made a collaboration with a US company called Amyris, who are specialists in engineering yeast into producing various different compounds. So squalene is actually on the path, natural synthesis path by eukaryotic cells to produce cholesterol, and therefore many cells can produce squalene. So they re-engineered yeast to produce the same squalene source in a more effective way. Thereby, we have established a more sustainable source of squalene, basically from fermented yeast. If you go to the next slide, I think I have an illustration of that. Yeah, you can see that we take the engineered yeast, we feed it with some sugar, let it ferment, and the yeast then starts producing the squalene. In this little micrograph here, you can actually see these are squalene yeast cells, and the small green dots within these cells is actually squalene. Then we take it, we purify it, and we can supply identical production of squalene, product of squalene, to what is extracted from shark liver. This is available in GMP now. Moving to the next slide. So these were just a few examples of how we take in and make available alternative sourcing of existing use. Of course, we also focus a lot on developing novel adjuvants, novel immune stimulators, novel adjuvant systems. This is, I think, this webinar is focusing on what's happening beyond alhydrogel and adjuvants. So I'll not focus much on those, but if you go to the next slide. I still want to mention that Croda has back in 2018-19 acquired the oldest commercial producer of aluminum adjuvants, producing the alhydrogel and adjuvants. So we have a long experience in working with vaccine adjuvants, both from, of course, from production point of view, but also from a regulatory and QA/QC level. What is required for adjuvants in the pharma space? If you go to the next slide. Yesterday, Derek also mentioned how important it is with these adjuvants to be able to formulate, and definitely alum. It's not necessarily a simple task to formulate vaccines with aluminum adjuvants. Therefore, there's a lot of physical chemical characteristics that can be optimized in association to it. And I think Derek very importantly highlighted this, that there's not a one solution fits all to how you formulate with aluminum adjuvants. And for sure, we can help in that aspect with our heritage products, with the alhydrogel and adjuvants products, to secure that they are used in the most optimal way. If you go to the next slide. But of course, the focus of this webinar is more what's happening beyond the aluminum adjuvants. And one of the platform technologies that we have taken into the Croda portfolio is the vaccine adjuvants from Statens Serum Institute. Dan mentioned there were two adjuvants from Statens Serum Institute in the CEPI library, CAFA1, and these are CAFA1 and CAFA9B, which we produce in Croda. We produce those and we can supply those in GMP. These two, I'll come back a little more into these adjuvants, but first mention that actually the CAF platform is more versatile than just these two adjuvants, and then there will be and can be more novel adjuvants coming out of that. The whole concept of it is that we have a platform, which is the cationic liposomes, that we can then combine with different types of immune stimulators that then in the end will generate different types of immune fingerprints. So move to the next slide. So the first generation is the CAFO1. CAFO1 has been used in a number of different clinical trials. There's plenty of clinical data on it, and quite good correlation between mouse and man in that aspect. It's a very, very simple composition. Maybe when you compare to some of the other adjuvants that are used today in clinics, for example, compared to the AS01, which contains the very complex QS21 you also heard about yesterday, the natural MPL, which where the whole production and accessibility to these can be quite tricky. These two components are very simple synthetic components that only that have only 2 to 3 synthesis steps each of them, and therefore the supply chain is quite effective, quite secure. They form a very stable adjuvant that are stable for several years, refrigerated, and the hallmark immune fingerprint is really a strong Th1, Th17 immune response and antibody responses that are at least, depending on the adjuvant antigen and what dose of antigen and all these things, but can at least be on par with aluminum, but have also been seen logs higher than with alum. Move to the next slide. Then another adjuvant, the other adjuvant we have taken in is the CAFO9B, which is and you could say an add-on to the platform. So in here we have replaced the TDB, the trehalose dibehynate used as mingle agonist in CAFO1 with a next generation one called MMG, monomucoglycerol. That targets the same receptor and actually induces the same downstream signaling and also the same immune fingerprint. So the DDA MMG formulation in itself generates much the same immune response as CAFO1, but then we add on the poly-IC immune stimulator, which is a TLR3 ligand. And thereby we also induce strong downstream type 1 interferon responses, facilitating, for example, CD8 T cell responses if the antigens are designed for that. This adjuvant is due to the fact that it targets TLR3 is really a very strong mucosal adjuvant, so can be used for intranasal administration also and induce strong IgA responses locally. This has not been tested in humans. In humans, actually, it has been used in cancer immune therapy, where it's actually been injected intraperitoneally, simply due to the same fact that it's in order to stimulate the strong CD8 responses, you need to target the cross-priming dendritic cells, and they are primarily associated with mucosal tissues, and the peritoneum is probably the best site for that. So in the cancer immune therapy, where it's feasible, actually the intraperitoneal immunization is really effective. Probably not the choice for an influenza vaccine, but here you could then choose the mucosal route. Next slide, please. So I mentioned the AS01 and Derek, of course, introduced it yesterday. That as as Dan also mentioned before, there is this lemming effect in the vaccine field, that if you have found an adjuvant that is really good, everybody wants it. And we also appreciate that, and definitely AS01 is an effective adjuvant, with the caveats it has that was also mentioned yesterday. We have made available and can make it available also in GMP now, an alternative to that, which is as close to AS01 as we can get. It is not AS01. We use a synthetic MPL analog. We use the 3D6 acyl fat, and of course it's not exactly the same QS21, since that QS21 is produced by GSK, but the concept of the liposomes are exactly the same, and the immune profile also the same. That can be made available, and we are also looking into making that available in the future. For example, through the CEPI catalog or in the NIH catalog. So next slide. And another platform, since we have the squalene and we have the excipients, of course, we can also, for now, for research use only, we don't have a specific strategy. Our strategy is really to support the squalene emulsion field, adjuvant field with our excipients. But of course, we also have in our research portfolio a number of different squalene emulsion adjuvants that we can make available. And then here we are primarily focusing on combining them with other TLR ligands. But we can make analogs available to basically all those existing adjuvants based on squalene, since we also have easy access to all the surfactants and lipids used in these formulations. Next slide. But I think what is important here is to say that over the coming period, in the last 5 years at least, but also over the coming period, it is maybe not so much the accessibility that is the major hurdle for many of you, especially in the research phase of your selection. It's rather what adjuvant should you then choose, and how should you use it. Move to the next slide. And I'll use the last, what do I have, 10 minutes on focusing on that, because there is not a one solution fits all. We know, yeah, everybody likes the AS01 or the ISCOMs right now, the Matrix M right now, but that doesn't mean that they are the perfect solution for all our vaccines. On the contrary, actually, because they do not fill some certain gaps of immune fingerprints. So what is really important, and that's also, I think, what I'm certain that what the CEPI catalog and the whole CEPI adjuvant library purpose is about, is to find the right adjuvant, not only the accessible adjuvant, but the one that fits best to the antigens. And that fits best to the correlates of protection. So, of course, we need to have a lot of things in mind with that. For example, how is the vaccination protocol done? What spacing between vaccines? And many of these things can be difficult to mimic in animals, but at least we can do attempts to make it better. Therefore, it's really important to understand what these adjuvants can do and cannot do. And here, of course, we have an enormous expertise in the adjuvants that we are working with to understand the pharmacokinetics, the mechanisms of action, and not least the immune fingerprints you get out in the end. Move to the next slide. I just want to give an example that I worked with a few years back, and I think Derek was very good at highlighting the schisma of how different adjuvants work, and that it's important to compare them. What I would like to illustrate here is also that you shouldn't just compare them under the same circumstances, but compare them adapted to having the adjuvants performing the best possible way individually, and then see what works. One study we made some years ago, we compared the CAFA1 adjuvant from SSI with the Adavax, which is really a mimicry and analog of the MF59. We gave one immunization of these, and then we focused on, of course, on the antibody responses, the T-cell responses, and the kinetics, the pharmacokinetics of the whole immune responses. If you go to the next slide. And what we observed, and that was not surprising, it's been published before, one of the qualities of the squalene emulsions is really that you very rapidly get a strong antibody response up. Already after a week, you start to see significant antibody responses, which grow over the next weeks. Where with the CAF adjuvants, which are really more they're forming a depot, they have a slower presentation of their, they have a quite opposite mechanism of action of the squalene emulsions when it comes to the pharmacokinetics. It's much slower. In the end, the magnitude of the antibody responses is superseded, so that CAF1 actually induces stronger antibody responses. But it takes time. So maybe not the adjuvant to choose for a pandemic where you basically want your antibodies before you inject your vaccine. On the other hand, you see very low T-cell responses with the squalene emulsions, where you do see strong both interferon gamma and IL-17 in the case with CAFA1, so Th1 and Th17 responses. After one immunization, of course, that declines. You really need to in order to establish effective memory T-cell responses, you need a booster vaccine. We didn't do that here because that would blur the whole thing. If you move to the next slide. But just to illustrate how different these two adjuvant systems are in their mechanisms, or in their pharmacokinetics, in their delivery properties, then we looked at the antigen-positive cells in the site of injection and in the draining lymph node, where we can see that the squalene emulsions, they're the blue dots, they very quickly, if you look at the bottom figure, very quickly come into the draining lymph node. This is not surprising. Novartis did a brilliant job in mapping that out much earlier than this paper. But where we do see a little, but very, very little antigen positive cells in the draining lymph node over time, and that's basically because it's stretched over a very long period of time. What, on the other hand, we do see is that there's plenty of antigen positive dendritic cells or monocyte cells in at the site of injection with the CAF adjuvant. So the whole reaction actually happens with the CAF adjuvant happens at the site of injection, whereas the squalene emulsions, a lot of the activity happens in the draining lymph node. Next slide. And that also has an effect on the germinal center formation. So if you look at the graph to the left, you will see that with the squalene emulsions, the blue dots, you see a very, very quick establishment of germinal center cells. And you see germinal centers being built up much faster with the squalene emulsions compared to the CAFA1. On the other hand, you also see a faster decline after a couple of weeks, you begin to see a decline with the squalene emulsions, where the curve is still up going for the CAF adjuvant. So the kinetics of the germinal center formation is also completely different. And it is still big with the CAF1 after, in this case, 42 days. But later studies have shown that you still see it after 80 days. You would still see a little germinal centers in the draining lymph nodes with CAF. So the kinetics of the germinal centers are completely different. Go to the next slide. And that's really important, because what implications do that have? I think most of people working with animal models know that the classical, maybe except when if you go into pigs or non-human primates, the standard regime is 2 to 3 vaccinations with 2 to 3 weeks interval. But we also all know that the most optimal time point to vaccinate or to come with a booster is actually when the germinal centers have collapsed. Then the immune system is more ready to take on a booster vaccine, which means that we actually need, for the different adjuvants or the different vaccines in general, we need to space our vaccination protocol according to that. We never do that in mice. Actually, even if we are stretching it to say that we give a vaccine booster at 3 weeks, we still actually just give it additional priming in the mice, for example, because the germinal centers are still alive and thriving with I would postulate 99% of all vaccines, if not more, meaning that we actually use our animals wrong. We should wait. Optimally, we should wait maybe 2 months, 3 months before we actually come with the booster vaccine. We know we cannot do that, but at least we can attempt to say, okay, for the different vaccines where that we know that there are different kinetics, we should maybe try to plan it so that we vaccinate more properly with the vaccines. At least we should have in mind that this is a factor. We saw during the COVID in the clinical development of the different corona vaccines, especially the mRNA vaccines versus the vector-based vaccines, we actually saw that they discovered that there were huge differences in the most optimal time points for the primes and boosts. This should be taken in and can be taken in much earlier in our selection, and should also be taken into consideration when choosing the right adjuvant, depending on what type of immunity you want to obtain. Next slide. Yeah, and just press one more time. Because I think what is stated here is we can help you to guide you to what adjuvants you should choose, depending on what correlates of immunity you want. But what is also really important is that you should go to those, if you're uncertain, you should go to those experts in the vaccine adjuvant field that work with these adjuvants and really try to learn how are they best used, because both when it comes to the formulation optimization, Derek also highlighted that yesterday, but also how should I design my preclinical studies to get the best out of these adjuvant systems, which will vary from adjuvant to adjuvant. And I think with that was my last important slide. I think yes. So if you have any questions, you're very welcome to contact me.
Yeah, thank you, Dennis. This was really helpful to understand what CRODA does and all the different adjuvants that you have. So I'll give it to Robert for the questions.
Yeah, great. Thanks, Lakshmi. And yeah, Dennis, thank you for an excellent presentation. So looks like we have time for a couple questions that I see in the chat here. So the first one is regarding the synthetic squalene And basically, if you have a sense of how this is going to be viewed by regulators, the example is given in India, where there's often a request for historical data, and the characterized squalene obviously has been used extensively. So, just if you have any comments on the regulatory acceptance of the synthetic versions, that would be great.
Yeah, so first of all, there are no products on the market with this, but it's but we are in quite good dialogue with regulatory authorities. The only pharmacopoeia existing right now that is the pharmacopoeia in EMA, and right now it actually states that the source is from shark. They are working on removing that because they see that as a problem. I think what we have experienced is that there's a very positive take up with this from regulators, because they recognize that it's not sustainable. So they need to be adaptable. What we can document from our side is that it is chemically identical products. Actually, we have more insights into the impurity. It is plus 99% pure, like the squalene you also get from shark, but we have more insight into the purity profile, and we have a more reproducible impurity profile of the last 0.something% of our products. So we have much better insight and much better control of that. I think from a regulatory point of view, they all recognize we need to shift. There will be a demand in the future. So therefore, regulators also are looking very positively also into replacing the squalene in existing products without having regarding it as new products. Of course, there will be change requests in the documentation, but not with big new clinical trials on it. That's not my interpretation.
Okay, great. Yeah, thank you. That's great to hear. And then I think we just have time for one more question. There's a question about Quill A and NanoQuil, and just understanding if they're available for commercial application, particularly in avian species, or if you have suggestions of other adjuvants which could be used in avian species.
Yeah, so Quill A, we have that as a commercial product and have had that for many years. NanoQuil, I didn't go much into it here. They're 100% for veterinary business. So the NanoQuil is you could say a next generation version. We use the same Quill A, but the problem with Quill A is that it is reactogenic. You basically inject saponins directly in. NanoQuil is a cheap developed method to reduce that reactogenicity by formulating the saponins together with cholesterol, and thereby reducing reactogenicity but maintaining immunogenicity. It's still a developmental product. It can, and depending on the markets, I would say it's relatively easy to get it into the non-regulated veterinary markets, which most LMIC countries are. If you want to move the products into regulated markets, it takes some more development, because we would need to produce it in GMP, and there we are, I would say, when there's a pull, we will push it into the GMP space, but that's not where we see it now. We do have alternatives also. That there's a lot of focus, of course, in the veterinary field for the mineral oil emulsions, where we also have some candidates in the non-regulated markets for now. That would be good replacements for the problem with the current, without going too much into detail, is really that they need to be emulsified at the site, in the praxis, where we have developed some mineral oil replacements, where you can basically just mix the antigen without the emulsification step, which makes it a little more easy to work with. that can be made available. Still, we haven't pushed them into GMP. But there is not, I'm very sorry to say, there is not much push into that field of novel adjuvants because price point is simply too low. So there's not much wiggle room to find good candidates that are more effective. And especially for the avian, of course, we have the problem that we know very, very little about, actually, surprisingly, about very little about the immune stimulation in birds.
Okay, great. Thank you for that. Okay, I think we should move on. So I'll turn it back over to Lakshmi to introduce the next speaker, but thank you again, Dennis.
Yeah, thank you so much, Dennis. This was really wonderful. Our next speaker is Wolfgang Littner. Dr. Wolfgang Littner is the Chief of Innate Immunity section of the Basic Biology, Immunobiology branch, Division of Allergy, Immunology, and Transplantation at the NI and National Institute of Allergy and Infectious Diseases, NIAD, and NIH. He oversees the NIAID vaccine adjuvant program, and his section manages the grant portfolio in the area of innate immunity and programs to advance adjuvant development. He joined NIAD's extramural program in 2008, and after working on conventional and self-replicating DNA and RNA-based vaccines and adjuvants for melanoma and T-cell lymphoma at the National Cancer Institute for a decade. He's become interested in DNA vaccines during his postdoc at the Walter Reed Army Institute of Research, and where he developed the first DNA-based vaccine for malaria. So with that introduction, I'd like to invite Dr. Wolfgang Littner to speak about his work and adjuvants.
Thank you very much, Lakshmi. So I'm Wolfgang Littner. I represent the vaccine adjuvant program at the National Institutes of Allergy and Infectious Diseases. And this is an effort to create novel adjuvants. It is housed in the extramural program of the NIAID, which is the funding arm of the Institute. And we work with academic labs and companies in the US and around the world to develop novel adjuvants. So it's a program that started in 2004. It was designed to address the issue of limited availability of defined, safe, cheap, and accessible adjuvants. This image here summarizes the ingredients of a clinical stage vaccine. And I like to show it to remind you, as Dan Fullen did earlier today, that ironically, from a regulatory standpoint, adjuvants are grouped together with other excipients. And that despite the fact that the choice of adjuvant can make or break a vaccine. You've seen this information packaged differently already in presentations yesterday and from Dennis Christensen right before me. It highlights how far vaccine adjuvants have come in the last 2 decades. So here on the left, you can see representative vaccines that were in clinical use in the early 2000s. And almost every vaccine that was based on an exogenous adjuvant contained an aluminum salt or alum, with one notable exception, and that is the squalene adjuvant MF59 that was part of a new flu vaccine approved in Europe in the late 1990s. And this landscape has changed dramatically since then. And what you're seeing here is a non-exhaustive list of a current list of licensed vaccines that contain novel adjuvants. 2 of them came out of our program. These are the combination adjuvants Advax CpG and L-hydroxyquim. The latter one, L-hydroxyquim, is based on the same principle as AS37 that's under development at GSK that you heard about yesterday. It's a small molecule TLR7/8 agonist that's absorbed onto alum to control delivery and availability of a very potent TLR agonist. Now, while this situation is a significant improvement over where we've been earlier this decade, but there are many, many more adjuvants in the pipeline. And the reason why we aren't seeing them in products or not even in late stage clinical trials yet is not that they aren't potent enough or not safe, but primarily because of the high costs associated with clinical trials. What I'm showing you here is a summary of NIAID's adjuvant program, primarily to put what I will be talking about in the next slides into context. We support adjuvant research and development from the discovery of brand new classes of adjuvants through preclinical evaluation, optimization, and down selection of adjuvants and mimics of established adjuvants. And then other divisions at NIAID support clinical trials of those novel vaccine candidates or vaccine candidates with novel adjuvants. One exception that I wanted to point to is a clinical program that we directly support, and that is the HEAL program, where novel adjuvants are used not for infectious disease vaccines, but for substance abuse vaccines. And this is just to highlight the broad usefulness of adjuvants beyond infectious diseases and cancer. Now, what are we trying to accomplish with our program? Developing a novel adjuvant isn't just about increasing or ever increasing immune responses, but also to induce the right immune responses. So we've been building a portfolio of adjuvants that selectively drive Th1 versus Th2 versus Th17 responses, since different vaccines require different immune profiles to be fully effective. This also includes adjuvants that actively suppress immune responses to specific antigens by inducing regulatory T cells, and those are being developed for use in vaccines against allergy and autoimmunity. And furthermore, we are interested in adjuvants that work in vulnerable populations. So that means newborns or the elderly who tend to respond poorly to many vaccines. We heard a little bit earlier today about choosing the right adjuvant, and that can be a really agonizing process. We support several programs where adjuvants are compared side by side with the same antigen. It's very similar to the approach that Dan Fullen outlined earlier today that CEPI is using. And this not only helps with the down selection process, but it also establishes immunological fingerprints of adjuvants with the goal of using this information in the future to better guide the adjuvant selection process, to make it a rational process. Now, the mechanism of action of adjuvants came up several times yesterday. While it's not a regulatory requirement, knowing how an adjuvant stimulates the immune system makes the regulatory process easier. It will certainly reduce the anxiety level of a vaccine developer who has chosen that particular adjuvant. Vaccine developers tend to shy away from adjuvants that do not have a clinical track record yet. This is one of the major reasons why alum continues to be the default adjuvant and the go-to adjuvant for many vaccine developers. Our program is aiming to help get novel adjuvants into their first human study and therefore establish their safety credentials. And finally, the major bottleneck in the field has been accessibility to adjuvants. We heard about several very advanced adjuvant formulations, especially yesterday, but access to them tends to be highly restricted. So our program is creating adjuvants that are accessible, that can be taken into clinical trials, so not just used for preclinical studies, but also product development. And this includes both novel compounds, but also mimics of established adjuvants. In terms of the mimics, these are the kinds of formulations that Dennis lovingly called the lemming effect. Now, you can learn more about our programs related to the NIAID adjuvant portfolio on this website. The QR code for the URL is right here in the corner, but don't worry about catching it now. I'll show it again at the end of my presentation. So Getting back to a question of adjuvant choice and pairing it with the vaccine, which vaccine actually needs an exogenous adjuvant? And what we're looking at here is the spectrum of vaccines, from live pathogens on one end to subunit and recombinant protein or peptide vaccines on the other end. And what's being reported over and over again is that virulence and reactogenicity more or less directly correlates with immunogenicity, meaning the least immunogenic ones are the ones on the right side here, being the clean proteins and peptides. They really depend on exogenous adjuvants to be sufficiently immunogenic and efficacious. However, I would argue that pretty much any type of vaccine can potentially benefit from the addition of an adjuvant. And yes, that does include live or attenuated pathogens. Now, while those pathogen-derived vaccines do already contain endogenous immunostimulators, such as LPS or nucleic acid, those may not be either enough to induce a protective immune response, and in all populations, but that response may not be appropriate qualitatively. So it may not be the right, most appropriate immune response. Furthermore, inactivated microorganisms are missing some of the immunostimulators that are associated with living pathogens and that allow hosts to recognize that there is an ongoing infection. And exogenous adjuvants can compensate for that loss of adjuvanticity. Now, we already heard a little bit about antigen design being an important aspect of vaccine development and improvement. So I wanted to take a moment to address the various approaches used to optimize a vaccine. So first of all, antigen design, which includes things such as stabilizing a protein in its pre-fusion configuration or multimerizing them, will certainly improve immunogenicity. We can also improve immunogenicity by delivering the same antigen through different vaccine platforms when you prime versus boost. As Dennis just pointed out, this is something that often gets overlooked. You can improve immunogenicity simply by increasing immunization intervals. We can deliver vaccines by different routes for the prime versus the boost. This does not only enhance the immune response, but it can also direct it to relevant tissues, primarily used for drawing immune responses to the lung for respiratory vaccines. Now, talking about routes, vaccines have traditionally been delivered to one of the least suitable tissues from an immunological standpoint, meaning the muscle. A few licensed vaccines are injected into the skin, which from an immunological standpoint is a very relevant tissue. The COVID pandemic has accelerated the development of intranasal vaccines, and there are also other technologies, some already quite advanced, to deliver vaccines more efficiently, such as transcutaneous patches. And then finally, vaccines can be drastically improved by picking not only the right adjuvant, but also the right formulation that is most suitable for this particular antigen adjuvant pairing. Now, the key message on this slide is that when you consider all of these variables, there is no such thing as a universal adjuvant formulation that works for every antigen, every delivery route, or every disease. Each one of those parameters can affect the hierarchy of suitable adjuvants and whether they are even compatible with the vaccine at all. So that brings us to the key question: how do you select an adjuvant? Now, in a perfect world, the adjuvant that you pick should induce the desired immune profile that is associated with protection against the disease target. Now, unfortunately, that's not known for many diseases. It should be easily accessible to the vaccine developers, easy to manufacture, meaning cheap. It should also be suitable for use in humans, because otherwise there's no point in using it if you can't go past preclinical studies. You should also consider your target population and parameters such as age or the immune status that is affected by additional factors besides age. Now, in the real world, we don't have enough data yet to be able to rationally select adjuvants for a specific vaccine. We heard about CEPI's effort to come up with AI models that will help in the future to rationally select an adjuvant for a particular vaccine. The current workaround is an unbiased adjuvant comparison, the kind we just heard about from Dennis, where you take several adjuvants through preclinical studies and ideally even clinical trials, considering that the hierarchy of adjuvants may be affected by the species that they're used in. And you do this in order to identify the most appropriate antigen adjuvant pairing. As I mentioned initially, we support several programs that do exactly that. And the reason that they're listed right here, the reason I'm pointing that out is that such comparisons were almost impossible to conduct in the past because of the limited number of accessible adjuvants. Now, this is no longer the issue. Now the limiting funding is, the limiting factor is really the funding, and no longer the availability of adjuvants that you can compare and down select from. I'll get back to the issue of specific adjuvants and the question of access in a moment, but I'd like to take a short detour to challenge our understanding of what an adjuvant actually is. Adjuvants are a very heterogeneous group of molecules and signals. What they all share is the final biological effect that they have on an organism, meaning the adjuvant effect, which is the enhancement of adaptive immune responses. And that can be mediated by a very wide variety of signaling cascades and immune molecules. Now, as was mentioned before, many traditional adjuvants are natural products that come from pathogens such as the LPS derivatives, such as fat, or from plants such as QS21. Now, in contrast, a lot of the modern adjuvants are primarily synthetic molecules. They're either derivatives of those natural products, or they're small molecules that are identified in unbiased high throughput screens. The bottom line is that anything that stimulates the innate immune system could be an adjuvant. So these are not just agonists of innate immune receptors, but other examples would include inducers of cell death, sources of energy such as laser light. All of those will can lead to an adjuvant effect. So when we think about the mechanism of action of an adjuvant, particularly of a receptor agonist of an innate immune receptor, it's important to keep in mind that these receptors do not act like switches. So in other words, they don't just have an on or off position. Instead, think of them as a rheostat. The agonist dose, agonist structure, formulation will all affect the strength of the downstream signal. And that signal is not just a linear signal, but actually can be very broad and complex and lead to a variety of downstream effects, as shown here in this example for STING agonists. Now, that's the quantitative effect of an adjuvant. But since an adjuvant triggers these multiple downstream effects, it means that we should also be able to affect those signals independently of each other. In other words, we should be able to change the quality of the overall response that an adjuvant induces. And in the next slide, I will focus specifically on those two downstream signals that are highlighted here. What I'm showing you next are some data from one of the groups in our adjuvant discovery program, the University of Chicago. They started using known partial NF-kappa B inhibitors as coadjuvants for TLR agonists that signal through NF-kappa B. And the objective here was to, on one hand, block the unwanted inflammatory signals that are associated with NF-kappa B stimulation, because this would address reactogenicity, which is, again, responsible for adverse events. But on the other hand, the goal is to also improve the immunogenicity of the vaccine with those coadjuvants. Now, as we heard yesterday, the better, meaning the stronger an adjuvant is, the more it is associated with reactogenicity and therefore adverse events. So the question is that really true, or can we uncouple adjuvant-induced inflammation from immunogenicity? Now, in this pilot study, peptide-based NF-kappa B inhibitors were delivered together with a CpG adjuvant. And while CpG by itself triggers the systemic release of TNF-alpha and IL-6, an NF-kappa B inhibitor can actually completely suppress those inflammatory cytokines. So that's great. That takes care of the reactogenicity. But what about immunogenicity? And maybe counterintuitively, suppressing inflammatory responses induced by an adjuvant can, either using this peptide-based or a small molecule-based inhibitor of NF-kappa B, increase both the immunogenicity, as indicated on the left, by significantly higher antibody titers, as well as the efficacy, here shown in a model of SARS-CoV-2 viral neutralization. So the next step in this program has been the systematic search for small molecules that either enhance or suppress NF-kappa B or IRF signaling that is being induced by a variety of TLR, NLR, or STING agonists in order to identify completely novel classes of coadjuvants that by themselves are immunologically silent, but are able to modify the immune response that is being induced by a PRR agonist. Some of them are specific for certain PRRs, but others work across the spectrum. So these are the ones in red that you see here that would suppress the in this case, NF-kappa B activity induced by a variety of these receptor agonists. So now we know that we can challenge the no pain, no gain dogma of vaccines and uncouple inflammation and immunogenicity. But what about that fine-tuning of immune response through selective modulation of downstream signals that I mentioned before? So again, as a reminder, recognition of a PRR agonist here, we're looking at a nucleic acid receptor, triggers a variety of downstream effects and signals, cytokines, upregulation of co-stimulatory molecules, and so on. And what we're seeing here in this experiment is 3 different small molecule coadjuvants of a CpG adjuvant. One reduces CpG-induced TNF-alpha release, one eliminates it completely, and one actually enhances it. However, the impact on IL-6 secretion of these 3 compounds is completely different, with all 3 suppressing the release of that cytokine. And this is just one example of how it is actually possible to fine-tune vaccine-induced immune responses to eventually adjust the immune profile of your vaccine for maximum efficacy and minimal reactogenicity. Now, getting back to the portfolio, I want to summarize in which directions we're going with our program. We continue to support the development of synthetic derivatives of known PAMPs. These include INI 2002 and 2004 that you will hear more about in the next talk from Jay Evans, or for example, the Beck adjuvants from the University of Maryland, which are TLR4 agonists that are discovered by transfecting various LPS modifying enzymes into bacteria to create completely novel types of LPS derivatives, or novel CpG oligonucleotides, such as CpG 55.2, which is the adjuvant in the COVID vaccine spicogen. What all of these have in common is that we know the original PEMP, we know the molecular structure, and then build upon that knowledge. Now, more and more of the novel adjuvants, as I mentioned before, are small molecules that are identified in high throughput screening campaigns. And one example would be the TLR4 and TLR7 agonists in FOS47 from UCSD. In this case, even the TLR4 agonist is a small molecule. The starting point for those novel adjuvants normally is a random compound library, and the screen may be completely unbiased. And so you don't really know which receptor you're targeting and what you're going to be finding. As I mentioned before, we're also trying to address the issue of limited access to adjuvants, especially those that are in advanced stages of development. And we're doing this by supporting the development of adjuvant mimics. Examples of such mimics are EVX-S from Enapret, which is an alternative to MF59, but just like the squalene that Dennis just described, this formulation is also based on a squalene from transgenic yeast rather than shark livers. So in other words, it's a vegan adjuvant, and that is an important consideration for certain vaccine markets. These mimics also include alternatives to AS01, such as the LMQ formulation from the Vaccine Formulation Institute, or ALF-Q from the Walter Reed Army Institute of Research, or the LIT4Q from PAI Life Sciences. So in this case, the starting point is a well-established adjuvant, but mimics are, this is an important aspect to keep in mind, mimics are not necessarily exact copies. In other words, they're not biosimilars of the original, but they mimic the biological effect of the original, and in some cases using similar but not the same kind of agonist. I mentioned NF-kappa B inhibitors as response modifiers used as coadjuvants, but there are many more such molecules that are identified either, again, by screening compound libraries or using already known, well-defined immunomodulators, but in a completely new context, meaning as coadjuvants. Now, finally, I won't have time to talk about another big category of adjuvants, and those are combination adjuvants. I mentioned a few already, for example, AdVac-CpG. What we're attempting to do here is address the fact that in case of an infection, the immune system does not get triggered by just one single innate immune receptor agonist, but an infection triggers multiple receptors. And when you mimic that through a combination adjuvant, you may be able to achieve synergistic immune stimulation. Now, getting back to the major point of my talk, access to novel adjuvant. So to help vaccine developers with connect them with adjuvant developers, we created the Vaccine Adjuvant Compendium a few years ago. The VAC is a complementary effort to CEPI's adjuvant library that you heard about earlier today from Dan Fullen. This is an online database that allows you to search for adjuvants based on parameters such as the mechanism of action, state of development, the model system, and antigens that they have been used with so far. And this database is not restricted to adjuvants that we supported, but anybody who wants to collaborate with vaccine developers is welcome to feature their adjuvant in this online system. The VAC allows you to create data sheets for any of the adjuvants listed. I'm using as an example here the squalene emulsion SWE from the Vaccine Formulation Institute as an example. Important features are this Each of those records includes a point of contact for anybody who is interested in using the adjuvant for their vaccine development program. Each adjuvant also has a unique ID number right here. This ID number links it to the vaccine ontology site Ontobee. This is a site where the record is mirrored and the adjuvant is shown in its appropriate ontological context. And with that, I would like to finish, show you again the website of the NIAID Vaccine Adjuvant Program, different funding opportunities, and also if you have any questions later on, here is my contact information. Thank you very much.
Great. Thank you so much, Wolfgang. That was fantastic. Let's see. So I think we have time for some questions. I haven't seen any in the chat yet. So maybe just to give folks a chance before while they're putting questions in the chat, I had one thing that I wanted to bring up, and this was around basically, and I think I asked at least one of the speakers about this yesterday, just around the prospects for mucosal adjuvants specifically, and you touched on this a little bit about the possibility, even in some cases, live attenuated vaccines, may benefit from an adjuvant. So if you could maybe share a little more or expand on that a little bit, that would be fantastic. Thank you.
So one of the limitations with mucosal adjuvants, and this was mentioned yesterday, is that you cannot necessarily use adjuvants that have been developed for and optimized for intramuscular delivery. You can't simply deliver them by the, let's say, intranasal route. What we have in our program is adjuvants that were either specifically developed for intranasal delivery. Example is the Nanovax NEO1. This is a nano emulsion specifically designed for intranasal delivery, or you may be able to adjust the formulation. Example would be, I mentioned alhydroxyquin. This is an TLR7/8 agonist. It's adsorbed onto alum, which means you cannot deliver it by another route than intramuscular immunization. The next generation of that adjuvant is called Sequim. It's more or less the same TLR7/8 agonist, but now on a biopolymer, which allows you to deliver it by any site. Turns out, well, we've traditionally been very worried about delivering into the nose. There was bad experience with a flu vaccine in the early 2000s with an adjuvant that should not have been used by the intranasal route. There were a few adverse events after the vaccine was rolled out, and that really put a damper on the vaccine community's enthusiasm for intranasal delivery. We've learned a lot since then, and what we have learned is that you can actually safely deliver TLR agonists by the intranasal route. We'll probably hear more from Jay in a second about the delivery of a TLR4 agonist intranasally. Formulation is everything, and it is possible to safely deliver even potent TLR agonists by the intranasal route. In terms of the use of adjuvants for live attenuated or even non-attenuated pathogens, like I said before, and as Dennis pointed out, as Dan pointed out, context is everything. There is no universal adjuvant, and you have to consider the characteristics of your vaccine. Just like with an RNA vaccine, you do not want to use an adjuvant that interferes with the activity of the life attenuated or non-attenuated pathogen-based vaccine, meaning you don't want to prevent it from infecting and making antigen. There are a new generation of non-inflammatory and in some case even anti-inflammatory adjuvants. The poster child, you can almost call it, is Advax. Advax is crystalline inulin, basically a crystalline sugar. It turns out to be an anti-inflammatory adjuvant. And adjuvants like that would not expect it to be interfering with antigen expression with the ability of a life or life attenuated pathogen to do its thing, meaning infect and make antigens and trigger an immune response.
Great, thank you for that, Wolfgang. I think there's just one other question I see in the chat, and this is about mimic adjuvants and basically understanding how mimic adjuvants influence their translational potential. So yeah, if you could share your thoughts on that, that would be fantastic.
In terms of translational potential, I assume the question is about product development, getting the adjuvant into a clinical trial. So again, it's important to keep in mind that an adjuvant mimic is not a copy of the original. So any of the AS01 mimics that I mentioned or that others have referred to are not exactly chemically the same entities as the original. Therefore, they are independent products. They're not under the same restrictions as the original. Now, the regulators are going to look at them as a new product. They're not going to be treated like just a copy of the original. Therefore, you're not building upon the regulatory background of the original adjuvant. So you can't just take an AS01 mimic, go to the regulators and say, well, look, this has been in Shingrix, this has been in RXV, this has been in Mosquirix, and therefore it shouldn't be a problem. It is, number one, a different product. Number two, you're pairing it with a new antigen, and therefore it is a new product. And that's one thing to keep in mind. It doesn't automatically make it easier to get it into a clinical trial, to get it past the regulators. It does make it easier when it comes to access, because now you're dealing with a completely new independent product, independent of the original adjuvant. So you're not under the same IP restrictions.
Okay, great. Yeah, thank you for clarifying that for us. Okay, I don't see any other questions, so I think we will move on to the final speaker. So I'll turn it to Lakshmi to introduce, our last speaker. Thank you again, Wolfgang.
Yeah. Our last, not the least, speaker is Dr. Jay Evans. He's the co-founder and chief scientific and strategy officer of Inimmune Corporation, which is in Montana. And he's also the director and center for translational medicine at the University of Montana. So before joining Inimmune, Dr. Evans spent about 16 years at GSK Vaccine, leading the discovery and development of novel vaccines and immunomodulators and advancing programs through IND submissions. So with that introduction, I'd like Dr. Evans to present his talk on adjuvants. Dr. Evans.
Great, thank you very much. Sharing my screen here.
Yeah.
You able to see that okay?
Yes.
Okay, perfect. Thank you. Thank you very much. Thank you for the opportunity to present here this morning in the US, or possibly this afternoon based on where our audience is coming from. I'm going to talk to you today about adjuvant systems that are available through both Inimmune as well as our partner SPI Pharma, who is helping us in global distribution and commercialization of several of these adjuvants. And the topic is relevant to today with what we're doing with PATH, is because our goal is to really develop adjuvants that are accessible, available, as well as effective. Again, I'm Jay Evans. I'm the Chief Scientific Officer and co-founder at Inimmune. I'm also a faculty at the University of Montana. I've been working in the adjuvant space for 25 plus years. So I'll give a quick introduction and a little bit of background to the SPI Inimmune partnership. Spend most of my time focused on INI-2002, which is a synthetic, kind of a third or fourth generation TLR4 agonist, and a variety of different formulations. We've heard a little bit about the formulations that are AS01-like or mimetics. One of these is, of course, is 2002 plus QS21, and some additional formulations that are available with this adjuvant system. Depending on the time available, I'll then talk a little bit about some expanded adjuvant portfolio, other agonists that are also available, GMP, ready to go to the clinic or already in the clinic, with some other TLR agonists. So a little bit of history on Inimmune. We were founded in 2016. A group of 15 of us left GSK at that time. We were part of their adjuvant discovery and development team, moved from Hamilton, where MPL is manufactured, up to Missoula, Montana, and started Inimmune. And so we're really an end-to-end adjuvant discovery and development team, from the medicinal chemistry, designing and synthesizing the novel adjuvants, through formulations analytics, and now through clinical trials. We have our lead adjuvant is actually just finishing up a phase 2 clinical trial. Actually just recently finished, and we expect the unblinding here in a couple of weeks. So we focused on toll-like receptor adjuvants, but we're also now jumping into STING, C-type lectin receptors, and other types of adjuvant systems to fill some of the gaps in the portfolio. And we really have 3 different pillars that we're working on. One is disease-modifying immunotherapeutics in allergy and autoimmunity. We have one, actually a TLR7/8 agonist that's a vaccine adjuvant, but is also in a phase 1 immuno-oncology study. And then the adjuvant side of the business, which has been partially partnered with SPI, and that is getting these adjuvants out to the developers who need them. And this is, as Wolfgang mentioned, this has been largely funded through the NIAID Adjuvant Discovery and Development Program. Really, when we were with GSK before, and now with the University of Montana and Inimmune, we continue to receive strong support from NIAID, which is greatly supported through this program. Kind of a view of the pipeline right now at Inimmune. Our lead asset is INI-2004. It's a synthetic TLR4 agonist given intranasally for seasonal allergic rhinitis. This is our lead mucosal adjuvant. It's a synthetic TLR4 agonist that's about 1,000 times more potent than MPL, about 100 times more potent than GLA, FAD, or 2002, and it's designated for intranasal delivery with a mucoadhesive liposome. It's recently completed a phase 2 clinical trial. We're actually going to have that unblinded here in about 10 days. Excited for the results. What we can say is this TLR4 agonist, given up to 500 micrograms in a person, is safe. We haven't unblinded it yet, but we know there weren't any AEs, significant AEs or drug-related AEs in the phase 1 or phase 2. So we're excited about that, and we're interested to see the efficacy. The presentation today isn't on mucosal adjuvants, but if people are interested in 2004 as a mucosal adjuvant, it's been fully de-risked from a safety perspective in people. It just simply need to add an antigen. We have a spray device and everything ready to go. It's a great mucosal adjuvant. 4001 is a TLR7/8 agonist that's currently completing a phase 1 study in oncology. It's also been approved for a phase 1 clinical trial for an anti-heroin vaccine. Wolfgang had mentioned the HEAL initiative earlier with anti-opioid vaccines. This is included in one of those vaccines that's funded through NIH to get through phase 1 clinical trial. We're expected to start that here in the coming months. And then the third pillar is really what I'm going to focus my time on today, and that's the vaccine adjuvants and the adjuvant portfolio that we have in combination with SPI and Qvant Biosciences. So I know it's probably 18 months ago now, we decided to form a partnership with SPI Pharma because they produce alum and other vaccine excipients. They have a global supply chain and partners all over the world. And so we partner with them to help get 2002 out to the developers who need it. SPI also developed a relationship with Qvant for supply of saponin products, and so together we're building a portfolio of adjuvants to provide accessibility and cost effectiveness, across the vaccine developers. And so what we've been able to do is leverage the strengths of commercialization through SPI Pharma, as well as the different saponins through Qvant, and then through Inimmune, our synthetic adjuvants, to come up with basically a toolbox of adjuvants for developers where they can come here and evaluate whether it's alum absorbed, ASL1 liposome, mucoadhesive liposomes, emulsions, all with 2002, or with a saponin based on what they're looking for, and a versatile platform that can be tested across a variety of different antigen systems. And so the compound that I want to talk to you primarily about today is INI 2002. It's what I call a fourth generation synthetic TLR4 agonist. And I'll show you, I think it's on the next slide. I'll just actually come to this one and jump back to the other one. This isn't an exhaustive list of the generations of synthetic TLR4 agonists that are out there, but it really follows our story, which started with MPL, starting with Rebe Immunochem. Everyone knows it's the component in ASO4 and ASO1 approved adjuvant systems. It's a mixture of congeners. It's a really good adjuvant system, but it has some complexity in its manufacture and characterization because of that. From this, there's a number of disaccharide adjuvants that have been developed. And you heard about GLA and FAD just earlier this morning. These are generation 1 disaccharide synthetic adjuvants. Generation 1 doesn't mean less effective. It just means that they've been around longer. There's also things like the BET compounds that would fall into this class that I would call, maybe generation 1, maybe you'll call them generation 4 because they're more recently developed, but they're all disaccharides. And then in the early days of Crixa Corporation and Rebe Immunochem, these were then subsequently modified to remove one of the sugars to generate this AGP series of compounds. And a couple of those, RC529 and 601 and 527 were all made GMP. A couple of those have been in human clinical trials. One thing that all of these disaccharides and monosaccharide compounds have in common is they have some issues with stability, thermostability, and that if you put them in an aqueous solution, especially if you heat them to room temperature, that primary acyl chain will fall off, limiting their shelf life to usually 1 to 2 years in an aqueous formulation of 4 degrees C. So when we started Inimmune in 2016, one of the goals of our medicinal chemistry group was to solve these stability issues. And they were able to come up with what we call the DAPs, and the classic compound there, 2002, is shown under Gen 4. They inverted the stereochemistry in that first sugar, and that phosphate was actually replaced with a sulfate. And so this compound is called INI 2002. And if you see this slide, the bottom left-hand corner, this is in an simple aqueous 2% glycerin formulation for a year at 40 degrees C. And in any of the formulations we've tested, now we're out several years with a couple of these formulations, it's completely thermostable at room temperature of 40 C in a simple aqueous solution or the API. Our most recent GMP lot of 2002 is now stored at room temperature. And we believe it's going to have 5 to maybe 10 years of stability of the API at those temperatures. And so that's really the big differentiator of 2002 from some of these other synthetic TLR agonists. Bottom right, you can see 2002 was designed to have equivalent potency to GLA, FAD, or MPL, so it could substitute into those formulations and have equivalent reactogenicity and relative potency. The compound I mentioned earlier, 2004, which is currently in phase 2 clinical trials as a standalone treatment for seasonal allergic rhinitis, but is also a great mucosal adjuvant, is about 1,000 times more active than MPL. And was specifically designed for mucosal delivery. And the nice thing about synthetics is you can modulate the relative potency. We have everything from TLR4 antagonists to things that are 1,000 times more potent than MPL or GLA, to really tailor the immune response to what you want. And so the other thing is 2002 is designed for formulation in mind, ease of formulation. So we can readily formulate it into liposomes, regular liposomes, mucoadhesive liposomes, emulsions, or absorb it to aluminum hydroxide. It actually goes into a simple 2% glycerin solution that then could be used for subsequent absorption to aluminum hydroxide or to test the adjuvant in the absence of a delivery system. When you combine this with squalene, alum from SPI, or with different sources of QS21, you come up with the formulation systems on the right. For T4Q, which is our liposome, including QS21, to T4SE, which is the squalene-based emulsion, we use about 10 times less squalene than MF59 or ASO3. In that, we actually looked at identified the best ratio for use with 2002. We have veterinary products as well as 2002 absorbed to aluminum hydroxide. So talk a little bit about our partnership with Qvant Biosciences, where we also have GMP material made with Desert King QS21, but we think that Qvant's process of generating QS21 using full biomass has the potential to reduce the cost and supply constraints around QS21. And with this QSAP technology from Qvant, there's actually 3 different products we're developing. One is the traditional QS21 Infinity, which is a high purity, 95% pure QS21. The second is a QS21 focused on global health, and I'll explain what that means here in a few minutes. It's really the cost driver for these vaccines is the QS21 component in these AS01-like liposomes. And then QSAP technology is a veterinary grade saponin. So what do those look like? You can see here the middle one, QS21 Infinity. It's a 95% pure QS21 peak. Similar product is available from Desert King as well as Qvant, and there's others that are developing this technology. That's the classic QS21 that's in AS01 and these other AS01 mimetic formulations. A little bit later in the talk, I'll show you a slide that has some costing information on it. That's the driver. That's the component that really drives the cost of these AS01-like formulations. So we worked with Qvant to come up with what we call QS21 Global Health. You can see it's a little bit broader cut of QS21 that greatly reduces the cost for purification and the product you get out. And I'll show you data here in a little bit. In our hands, we've seen equivalent efficacy and safety with this product. And finally, on the bottom is the veterinary grade QSAP. As you can see, that's a much broader cut that greatly brings down the cost of that product for veterinary use. And so I'll start with T4Q, and that is our equivalent of you could say the AS01 mimetic design with QS21 and 2002. We're simply replacing MPL with 2002 and using this. And we've already made GMP lots of this. They're available now to go into a phase 1 clinical trial. As far as comparability, this is directly comparing AS01 and T4Q in an influenza vaccine model. As far as Th1 response, you can see interferon gamma, they're equivalent. IL-5, actually the T4Q reduces IL-5 a little bit more than AS01 does because 2002 is a little bit more stronger Th1 bias than MPL. The IgG2A responses are similar as well as the IgG1 responses are similar, which makes sense because 2002 was designed to have the equivalent potency to MPL. Chemical stability is where it shines. You can see here at 12 months, T4Q, good chemical stability, great colloidal stability. We have stability now out 2 years. Again, the limiting factor in stability here, this is stored at 4 degrees C, and not because of the 2002, but because of the QS21 and the lipids that are in the liposome can't be stored at 25 or 40 degrees C. But we expect we're going to have 2 years plus storage half-life or storage life of this GMP product. Again, this is already made GMP and available today to customers. And then if you look at this, the performance of this in an influenza challenge model, these mice were vaccinated with an A Victoria strain from 09 and challenged with a 1968, the H3N2 strain, so a fully drifted strain. There's no cross-reactive neutralizing antibody or HAI. We believe this is probably T-cell mediated. And you can see that when you give T4Q with the influenza vaccine, you get 100% protection, whereas the antigen alone, like the naive, because there's no neutralizing antibody here or HAI cross-reactive titers, all those mice will succumb to the infection. Another model system I'll show you about, especially when we're talking about developing adjuvants for LMICs, we worked with Pat Duffy at NIH on this malaria vaccine model for a transmission blocking vaccine. And I like showing this slide because it shows the variety of formulations. And with this specific transmission blocking antigen, all of these formulations worked well. It isn't always the case with every antigen, as Wolfgang and others have mentioned, but we looked at simple aqueous formulations of either 2002, which is our TLR4 agonist, 2004, the middle one here, which is our TLR7/8 agonist, or the two in combination. S&P, this is actually a Minkel adjuvant that I'm not going to talk about today, but also is available. It's not made GMP yet, so that's why I'm not talking about it today. Emulsion formulations of these, and then liposome formulations of these compared to AS01 on the far right in the light blue. And you can see all these adjuvant formulations do a really good job of driving high antibody titers against this TBV antigen from malaria, and then they block oocyte transmission in the mosquitoes. With actually this Minkel agonist performing really well, as well as T4Q, our version of AS01, also outperforming the others as far as blocking. And this is really just to show the versatility of these adjuvants across the different formulations. If you were to look at a different antigen, you might find that one of them really likes emulsions, or something you want to get a strong T-cell response, the liposome might be the better choice. And Wolfgang just talked a little bit about adjuvant selection and the complexity of how you mix that with the various antigens. So now I mentioned this T4Q global health. This is with the version of QS21 that's a little bit broader cut, which significantly brings down the cost. And is really targeted for LMICs in global health areas where the cost of QS21 at 95% pure might be a restricting component of developing that vaccine. So here we simply compared the global health version of QS21, the little bit broader cut, with the QS21, the 95% pure. You can see here for IgG1, they're equivalent. For IgG2A, they're equivalent. And then for interferon gamma, they're roughly equivalent. Might be a little bit less in the global health QS21 than the other. And we didn't see any safety differences in the animal models we've been tested to date. So I think this is a good option for vaccine developers that are cost sensitive. And I'll actually show some cost comparisons a little bit later what that looks like. So the third system here is T4SE, and that is 2002 with a simple squalene emulsion, which is less expensive to manufacture than a liposome, and of course doesn't have QS21 in here, so it also reduces the cost from that perspective. We did do a full development on the ratio of 2002 to squalene and found that we can use about a 10 times lower dose of squalene than is found in AS03 or M59 and get a better response than those adjuvant systems without the TLR4 agonist. We published a paper here in 2024 on SARS-CoV-2, and I'm going to highlight a little bit of the data because it really shows the benefits of this 2002 squalene-based formulation. And again here, similar to the other, the blue here, anything that's in blue is the formulation that contains our TLR7/8 agonist 4001. In red is 2002 containing emulsion, and in purple is the combination which we call TRAC of the 2002 and 4001 together. ASO3 like formulation is in gray, and then the Adjuvants is on the far right on the other side on gray. And you can see when you add all of these emulsion formulations do a great job of generating antibody to, this is the spike trimer from the Wuhan strain, came from the group of Mount Sinai with Ford and Kramer. When you look at IgG1, you actually get lower IgG1 because it's these are such strong Th1 polarizing adjuvants that you derive higher IgG2C and a lower IgG1. This can really be seen in the T cell response and the T cell recall here. In colors, of course, you got 4001 TLR78, in salmon is 2002, and then purple is the combination. If you look at IL-5 on the left, anytime you add a TLR agonist, it takes that high IL-5 response that you get with these squalene-based emulsions and basically drops it to almost zero, because these are such strong Th1 polarizing adjuvants. On the right, you can see interferon gamma. Anytime you add a TLR agonist, whether it's TLR4, TLR78, or the combination, you drive the interferon gamma response. And then when we looked at neutralization titers against either the parent Wuhan strain or Gamma or the Omicron variant, we found that this 2002 emulsion that's circled here in green was actually uniquely was able to neutralize the Omicron variant in mice that were vaccinated with the Wuhan. It worked better than 4001 or the combination, which was surprising to us. This is, as Wolfgang says, sometimes you have to test to figure out which adjuvant or which adjuvant system an antigen seems to like the best and drives neutralization. And when we challenged the K18 mice with either the Wuhan virus or this Omicron XBB1 variant, the Wuhan is expected, all the adjuvants protected, it's neutralizing antibody response, and even the Adjuvants or ASO3 like emulsion all protected really well. But when you then challenged instead with the XBB1 Omicron variant, and just like we saw in the past slide with the neutralizing antibody titers, this T4SE with a spike trimer there in salmon across the top, these mice didn't lose any weight and they were completely protected. So some of these adjuvants seem to have the ability to drive cross protective neutralizing antibodies that will cross between the Wuhan variant and the current circulating Omicron strains, which is exciting to the thought that you could maybe develop a COVID vaccine that is super seasonal and will protect against variants. So finally, T4AH, this is 2002 absorbed to alum. Like the SE formulation, this is going to be highly thermostable. This will be a vaccine adjuvant that doesn't have any lipids in it, doesn't have any QS21 or cholesterol in it. And so it's going to be stable at room temperature or even 40 C, completely avoid cold chain if the antigen also that's combined with it can avoid cold chain. And like I said, we can fully absorb 2002 and the antigen here we're using RBD to alum. So you basically get 95 to 100% absorption of both the 2002 and the alum. It's this way with most antigens. You have to have about 1.5-fold excess alum by weight to get full absorption, and you can usually get away with it using a lower alum dose than you would in a traditional vaccine. And again, this is 40 degrees C. This only goes out to 4.5 months, but we expect this is going to go out years. Even at 40 degrees C, the formulation is going to be completely stable because 2002 just doesn't seem to break down as thermostable. And then we made engineering lots of this already, so all the test specifications are set and ready for GMP manufacturing. And so just a little bit on performance of this. This is an RBD vaccine here, based vaccine from SARS-CoV-2, again, the original Wuhan strain. A little bit different than the full-length spike antigen I showed earlier. Looking at IgG titers, antigen alone with RBD, you add 2002, you get IgG. Here you add alum, it boosts the response, but not as much as the adjuvant. And then the two together here give an equivalent response to the 2002 alone. IgG2, again, you can see the Th1 polarization seen here in the antibody response with either 2002 alone or 2002 on alum. And then uniquely with this antigen, which I couldn't have predicted that the 2002 aqueous version wouldn't have given a good T cell response, here the combination seems to drive a better gamma response than either adjuvant alone in a recall response. We get a lot of requests about conjugate vaccines. And some conjugate vaccines are absorbed to alum, and so we tested this here with it's a peptide conjugate vaccine in this case, 2002, and this is a peptide conjugated to CREM. 2002 an alum over here, again same type of scheme, whereas in the red is the CREM conjugate alone. You add alum, you could boost the antibody response a little bit. You add 2002, you boost it more, and then the two in combination are on the far right. And this vaccine works strikingly well following a single vaccination. As you can see here on the left, this is a dose response of CREM with a fixed dose of 1 microgram of 2002. You can see even as low as 0.1 micrograms, you're getting a really high antibody response, almost a maximal antibody response with just a single vaccination. It also demonstrates dose sparing. Because your antigen alone at 10 micrograms is giving you nowhere near the response, significantly lower response for antibody than your antigen adjuvant combination, where the antigen's only at 0.1 micrograms. So you get 10, maybe even 100-fold antigen dose sparing. So we think some of these adjuvants will actually be cost savings for adjuvant developers, because the most expensive component to this vaccine would be the antigen. In this case, it won't be the adjuvant. So it might be considerably more cost effective to use the adjuvant versus using the antigen alone. Of course, post-secondary, the groups tighten up. We didn't get a huge amount of booster response with the adjuvant because post-primary, the responses were already so high, but you can see the antigen alone is starting to come up a little bit with the booster response. But you can still see the significant, probably 100-fold or more, maybe 1,000-fold antigen dose sparing that we can get using this alum vaccine. So this is a slide I mentioned earlier that has some costing on it as well. And this is our current estimated commercial scale pricing that we expect we're going to be able to get to on commercial scale. And you can see on the left, so all of these are either GMP ready or already GMP manufactured. The only one that's not is this T4E vet. It's still in development, but we're really close on that one. You can see T4Q, our estimated cost per dose, per human dose, is going to be over $3.50 per dose. Whereas if you go to the T4Q Global Health, it's $1.50 a dose. That's just the price difference between the Global Health cut of QS21 versus the 95% pure QS21. When you eliminate QS21 altogether from the vaccine product, suddenly you're under 50 cents a dose for either the alum absorbed or the squalene emulsion version. And these are for fully vialed, prepared vaccines ready to go. And we expect at commercial scale, we will be below 50 cents a dose. And what enables that is our next batch of 2002 is probably going to be 10 kilos. Which enables these costs. In the veterinary vaccine space, we think we can get that below 10 cents a dose. Because those formulations will probably be vialed in bulk rather than in individual vials. So this gives the vaccine developer some perspective on what costing will look like at scale when these are fully commercially available and out there at scale and a licensed vaccine. The other thing is we're also selling the 2002 API itself, GMP, to developers that have their own formulation capabilities. We have some vet vaccine business where they have special, they use mineral oils and other types of oil and water systems that are also compatible with 2002. They may want to source their own QS21 and make their own AS01-like formulations. We have a number of vaccine developers that are just purchasing the 2002 API which is going to be room temperature stable for years, and that to provide additional cost savings when they want to do their own formulation, fill and finish in their own development facilities. And so, really the partnership between SPI, Inimmune, and Qvant is made available. Really these T4Q-based systems, the veterinary-based systems, as well as the T4SE emulsions and T4AH. 2002 is a patented compound, so developers will need a license to commercialize it. But the goal is to do non-exclusive licenses and make it broadly available and cost effective. Sometimes people are scared away about saying you need a license because they've had horror stories in the past about how expensive the licenses can be. And the goal of SPI isn't to make a license unavailable to most vaccine developers, but instead you need a license because it's patented. But that shouldn't be a barrier to development or a cost barrier to development. The goal is to provide, a cost-effective solution, make these things on large scale, and get them out to the developers in places that need them at a low cost. The idea here is scale, which brings down the cost, which makes it available to everybody at a costing that makes sense. And so with that, the last couple minutes here, I'll talk about some other adjuvants that are in the Inimmune portfolio, not currently partnered with SPI, but we believe that's the path they're probably taking. We've been working on adjuvants at Inimmune, since we left GSK for the last 10 plus years, and we have a portfolio of adjuvants. We just heard about the TLR4 and saponins in the middle. Talked a little bit about our TLR7/8 adjuvant that's currently in a phase 1 clinical trial, which is more Th1 polarizing, as you saw from the data with the spike trimer from the Wuhan strain. We also have a Minkel adjuvant called 1098 that is under a development contract with NIAID, which we expect will have GMP material for that here in the next couple of years. Not talking about it today because it's not GMP ready today for developers that people want to put in a phase 1 clinical trial right away. Couple of words on our TLR7/8 adjuvant called I-94001. It's been made GMP. It's already been through a phase 1 human clinical trial for oncology, and we're starting a phase 1 clinical trial here in the next couple of months on an anti-heroin vaccine, which I'll show you a little bit of data from. As Wolfgang mentioned earlier, there's a HEAL program funded through NIAID for developing adjuvanted anti-opioid vaccines. So we've developed an anti-fentanyl and an anti-heroin vaccine with our partner Marco Pravatoni from University of Washington and Sandy Comer from Columbia. It's basically a creme conjugate vaccine with either a hapten, either being fentanyl or heroin, with 4001 as the adjuvant on alum as a particle. Just to show you really quick how well these work. This is in mice looking at IgG1 or IgG2. So you have the creme conjugate alone. This is with the fentanyl or F1 hapten. On the left, you add alum, you can boost the response a little bit. You add 4001, you boost the response a little bit more. You add the two together and you get the highest antibody response. If you look at IgG2, it drives a really high IgG2 response, which IgG2 antibodies seem to be particularly well suited for blocking fentanyl from getting across the blood-brain barrier. One thing that came as a surprise, which we didn't expect, is we actually get increased antibody affinity. Not just higher titers, but irrespective of titer, the antibodies that we're generating when we add the adjuvant have higher affinity. Which we're trying to understand now what's going on in the germinal center and clonality to generate higher affinity antibodies, but we see that with this product. And here in rats, we have the same data in mice as well as in pigs. If you vaccinate rats with this vaccine, here with the antigen alone on the left or the antigen plus adjuvant on the right, if you look at anti-fentanyl antibody titers, you can see here in rats, you get higher titers. If you look at brain levels of fentanyl after challenge with fentanyl, you can see high fentanyl in the brain of rats that aren't vaccinated. And then you have antigen alone. And then when you add the adjuvants, the fentanyl is simply not getting to the brain of these rats. And if you look at O2, animals that got to below 50% O2 saturation after challenge, you can see the rats that received fentanyl and vaccine with 4001 are fully protected. And where this is at today, we actually have IND approval from the FDA for a heroin vaccine in the US with the same exact adjuvant system. That's going to start here in the next couple of months. And we've completed all of the GMP manufacturing, GLP tox to move the fentanyl vaccine to phase 1 as well. And I'm want to be cognizant of time. Just one final thing. Wolfgang had mentioned the TRAC 478 adjuvant system, which is simply a combination of 2002, the TLR4 agonist I told you about earlier, with our TLR78 agonist 4001. This combination can be used as in an aqueous, alum-absorbed liposome or emulsion formulation. And again, all the components are made GMP. We've actually had the emulsion formulation has been manufactured GMP funded by NIAID through the Civics program, and is targeted for a phase 1 clinical trial, hopefully in 2027 through that program. A little bit of data from Mount Sinai on this. It's their antigen. A lot of the data has been collected by them. This is a neuraminidase antigen, N1-MMP, developed by Florian Kramer. You can see here when combined with this TRAC, either aqueous, liposome, or emulsion. This is the cross-reactive NA inhibiting antibodies against either New Caledonia or this bald eagle H5N1 strain, and this was a vaccine strain from a Michigan, so it's a cross-neutralizing study. You can see when we add the adjuvant, we get cross-neutralizing antibodies in any of the formulations. The emulsion seems to work the best, and when you further diverge this out to the H5N1 from a bald eagle strain, again, you can see these emulsion formulations seem to be particularly well suited for cross-neutralization with this antigen. And then you can do challenge in mice, again, with vaccinated with this N1-MMP plus TRAC, weight loss on the top or survival on the bottom, which we can focus on. Here, the emulsion formulations in dark blue or purple, these mice are protected from challenge with these drifted strains of either H1N1 or H5N1 after vaccination with this N1-MMP. We have similar data with the H3 trimer, again provided by Florian Kramer's group at Mount Sinai, comparing either the Adjuvants or our TRAC78 emulsion formulation. Simple antibody titers here. This is again with this H3 trimer antigen alone. This is actually here as a liposome formulation or an emulsion. It works the same with IgG titers. The strongly Th1 polarizing, you can see the IgG, strong IgG2A titers, and then the interferon gamma response that's provided by adding the TLR agonists versus Adjuvants alone, which is just the squalene-based emulsion. And then in a flu challenge, again, this is a drifted flu challenge, like I showed earlier, whereas the we're using an H3 from a 2014 strain, and that this is a 1968 challenge strain. So there's no cross-neutralizing antibody or cross-neutralizing HAI. You can see the mice that were vaccinated with the TRAC 478, 100% survived challenge and varying degrees of survival with the Adjuvants or the antigen alone. There is weight loss in these mice after the challenge. They all go on to survive. And we believe this is probably indicative of a T cell response because, as I noted earlier, there's no cross-neutralizing antibody here. So we believe this is probably a recall of T cell response here, and then these mice are able to overcome that challenge and go back to 100% survival. So that was that. I'll wrap it up with that. So thanks for your time. As I noted, all of the adjuvant systems I described today are all GMP ready. They're all, the licenses available non-exclusively in all territories, all regions, all uses. Available to any developer that's interested in these vaccine adjuvant systems. So thank you.
Excellent, Jay. Thank you so much for a fantastic presentation. That was great. Really appreciate that. So we have a few minutes left for questions. And I think we have a few that were in the chat and that were not addressed. But Jay, maybe one thing I just wanted to follow up on you with while we're giving other people a chance to put their questions in. So you mentioned the intranasal mucosal formulations or the intranasal mucoadhesive formulations. I was wondering if you could say a little bit more about those, and then also, is there similar, or is that formulation potentially applicable for other mucoadhesive delivery for other routes, including either oral or respiratory, or is that kind of specific for intranasal?
Yeah, that's a great question. So we've developed quite a few mucosal formulations with different mucoadhesives and permeation enhancers. The mucoadhesive formulation that we have in phase 2 is a liposome-based formulation that's mucoadhesive with our TLR4 agonist 2004. It also works really well with our TLR7/8 agonist 4001. Is being manufactured again under Civics for a potential intranasal vaccine with those antigens described as Florian Kramer. So we have done intranasal. Of course, in mice, we've done intrapulmonary, although we believe from a safety perspective, we'd like to keep it in the nose. If you give these TLR adjuvants in the lungs, you will get some weight loss and ruffling in mice or other animals. So we think the strict intranasal dosing is much safer, and we've demonstrated safety again in phase 2 in humans with that. We've also done a number of sublingual studies. And it's effective also sublingually. We're actually developing food allergy treatments sublingually using the same formulations, and hope to get those to phase 1 here in the next couple of years. So as far as oral or pulmonary, the TLR agonists, I think if someone carefully stepped through clinical trials to figure out the dosing for intrapulmonary delivery, it would be possible, but I think that would be it would take some time to work through the regulatory challenges there. From an oral perspective, these TLR agonists we're talking about would need to have some sort of a, some sort of a enteric coated capsule to get them through the acidity. We haven't developed that yet, but we're talking with developers that have the technology to look at using these TLR agonists as oral vaccines by enterically coating them and getting them to where they need to be in the gut.
Okay, great. Thank you for that. There's another question actually from Lakshmi regarding potential for use with RNA-based vaccines. So if you have any thoughts on that topic, that would be great.
Absolutely. We have an active program. As I mentioned, I have a faculty appointment at the university. We have an active program looking at mRNA vaccines. We have tested 2002 and 4001 in a variety of different ILMPs with a number of mRNAs. 4001, as you might expect, drives a lot of type 1 interferon. You drive type 1 interferon, you shut down protein expression. It actually reduces antibody versus the antigen alone. 2002 is effective in the right type of ILNP. And we're continuing to work on that formulation to see if we can find something that's a clinically viable path, drives high enough antibody titers in combination. We presented a little bit recently on some genetic adjuvant work. It was at World Vaccine Congress. We actually have some genetic adjuvant work where we're expressing cytokines along with the mRNA as a separate mRNA. And those, we've seen some really good promise there. Our colleagues at West Virginia University, as well as Dave Dowling at BCH. There's a number of groups that have published now on expressing genetic adjuvants as an mRNA along with an antigen. We've seen some really good responses there. We're looking forward to where those might go. But again, those I didn't present on those today because they're still in early discovery, early to mid-stage discovery. They're a little ways away from GMP and GLP tox and the kind of things that developers are looking for now for the later stage systems they're wanting to license.
Great. Yeah, thank you for that. That makes sense. And Jay, just one other question, going back to the fentanyl vaccine. So it looks like the initial antibody responses you get are great. I just wanted you to comment on the durability of those responses, because I mean, certainly I'm imagining, down the line in clinical use, these may be, let's say, difficult to reach populations. So you'd want to have maximize the duration of protection. So any thoughts on that aspect of that approach?
Great question. We get that a lot. And to be honest, with the fentanyl vaccine specifically, beyond some of the pig studies and stuff where we've carried out, 3 to 6 months, we haven't answered the question on durability. With the anti-heroin vaccine entering phase 1 here this year, we'll hope to get that durability question answered in people. Of course, it's hard to do it in animal models at times, especially in mice. They just don't survive long enough. And the larger animal models can be cost prohibitive to carry them out for years. So we'll hopefully within a couple of years, we'll be able to answer that question on durability. Usually when people ask, I refer them to the polysaccharide conjugate vaccines. A lot of them are creme conjugated. A polysaccharide is a hapten. Fentanyl is a hapten. I would expect that we're going to get at least as long-lived antibodies as the conjugate vaccines with the polysaccharides. We might be able to enhance that a lot with our TLR agonists, especially with 4001, but we'll answer that clinically, hopefully here in the next couple of years. But great question, and we just don't know at this point.
Yeah, great. Okay, yeah, thank you for that. And then looks like Lakshmi had one other question, just going back to mRNA, if there's strategies for prolonging the response, and obviously that's been, a question for other mRNA vaccines.
Yeah, we actually have those studies ongoing now with the genetic adjuvants that I talked about. Because those genetic adjuvants work so well, and we can easily model in mice. You can see in the mRNA vaccines in mice, especially with the COVID antigen, you can see that drop off in antibody titers happen within 3 to 6 months. So it provides a good animal model system. So we're testing that now. Just look and see if those genetic adjuvants increase the durability, which is a critical question for the mRNA vaccines, as we all know.
Okay, great. Perfect. Thank you. Okay, I think that covers all the questions that we've seen. So I know we're just about out of time. So Jay, thank you again for a great presentation, and I'll turn it back over to Lakshmi for closing us out.
Yeah, thank you, everybody. Daniel, Dennis, and Jay. I think we had a very good talks today, talking with the different adjuvants, and help us to walk through in the future, for especially with the LMIC groups, because now we can see that there is so much choice, and there is a lot more to do, and we all know that. But at least we can see that there is a lot of possibilities. So with that, we'd like to close, and a reminder that we will be sending a short survey to all those who attended, and we welcome your feedback on the webinar. If there are any questions, please do come back, and you can even send it to the speakers directly, or to send it to us, and we will field it to the speakers. But again, many have asked about the presentations and recordings. So this definitely will be online in June on the PATH website. So again, I'd like to close by saying thank you to everybody. Thank you to all the speakers. It has been really a wonderful experience for all of us to be able to listen to all of you. Okay. Thank you.